Good afternoon, everyone, and welcome to all the participants to this first meeting of the series highlighting organic chemistry in Europe. In my role as the Italian representative for the Società Chimica Italiana in the board of EuChemS Division of Organic Chemistry, which I've had the privilege and honor to serve as the president for the past 3 years, I would express my gratitude to all the committee about the decision to start the series with highlighting organic chemistry in Italy. Building up the identity of a community involving European organic chemists, also including those societies out of Europe which belong to EuChemS, is the main aim of the society. And this is based on sharing excellence in academic and industrial research, as we are doing today. And this is also the aim of many other activities of the division, including the Young Investigator Workshop and the new awards for research, for service, the Young Investigator Award, and the award Female Organic Chemist of the Year. So it's going to be an exciting afternoon of excellent Italian organic chemistry, and I'm delighted to pass the word to our president, the president of the Division of Organic Chemistry of the Società Chimica Italiana, Professor Valeria Dauria, wishing all of you to enjoy the meeting today. Please, Valeria.
Thank you, Gianluca. Welcome everybody, also from my side and on behalf of the board of the Organic Chemistry Division of Italian Chemical Society. Many thanks, Pat, for this so interesting initiative and for selecting our country, our division going first in the series. In my introduction, I'll give you some facts and figures on organic chemistry state in our country, starting with undergraduate education. Of course, organic chemistry is a core subject in all scientific programs, and these figures only refer to bachelor and master classes specifically ascribable to chemistry discipline areas such as biotechnology, chemistry, cultural heritage, material chemistry, pharmacy. In Italy, we also have a pharmaceutical chemistry program that is quite uncommon in Europe. As you can see, there is a very wide diffusion at the territorial level of chemistry classes. And after a crisis period, we are now experiencing a positive trend in terms of graduate number. The employment rate is high, at least if compared to Italian standards, and the majority of employers are in the industrial sector. Indeed, Italy is the third chemical producer in Europe, with a share of total production of 10%, and it is also the 10th chemical producer worldwide. And with reference to some branches, Italy holds even higher positions in international rankings. For instance, as far as pharmaceutical active ingredients are concerned, Italy boasts a global leadership. There are about 3,000 companies active throughout the national territory, employing 180,000 highly qualified people. The production, industrial production, is organized in districts or clusters, and Lombardy cluster here in North Italy is the bigger one in terms of production workers, and it is also ranked among the top 5 regions in Europe. Let's turn attention to academic setting. Our ministry promotes highly interdisciplinary PhD programs, so it's not possible to extrapolate the actual number of PhD students enrolled in organic chemistry projects. These are the figures of official academic staff according to career stages, and our community is not so big, about 1% of total staff, but it boasts a high reputation within the academic community. So many organic chemists acted or act as head of the department where they work, including 11 departments that recently received the seal of excellence from our ministry. Other colleagues are deputy rectors or delegates for the research or all the other management position. There is a good track record of granted projects. We will see some examples in today's program. And our research, organic chemistry research, reflect the interdisciplinary nature of modern scientific research, with peaks of national and international excellence in several fields, to cite but a few: material chemistry, supramolecular chemistry, of course, development of modern synthetic organic chemistry method with a focus to green chemistry, carbohydrate chemistry, and chemistry with natural products. Our division, this is the board in charge. Many thanks to all members for the support in the setting of today's program, particularly to Professor Alessandro Abbotto, who will share with the PET the discussion moments in this afternoon. Our division with 800 members, 1/3 of them of young people under 35 years age, is the biggest division of the Italian Chemical Society. We promote many educational coordination, dissemination activities. I have no time to go into details, but at the very heart of our activities, the organization of schools. We have 5 natural schools covering different subjects. Physical organic chemistry with the European Winter School of Physical Organic Chemistry, industrial chemistry with the International School on Process Chemistry, organic synthesis with the International Summer School on Organic Synthesis. This school is also known as Cobella School, is very famous. It is the oldest in the group. Natural products with the International Summer School of Natural Products, and multidisciplinary organic chemistry with the EuChemS Advanced School of Organic Chemistry. With a tradition of excellence, our school attracts many students, young researchers from different countries. So I invite all young people in the audience to check these websites. The schools are also announced in EuChemS portal. Please attend at least one. Our division manages several awards to acknowledge significant, outstanding scientific achievement also by young people, people working in academia, in the industry, as well to the 3 best PhD theses. Also in this case, the call is open also to foreign scientists, providing that they are members of our Italian Chemical Society. In conclusion, our community is a vibrant community. Our meetings, you can see the picture of the last 2 meetings. Our meetings are always a special occasion of unforgettable experience of shared science and nice conviviality. I'll introduce now the today program. We have tried to offer an overview of course not comprehensive, of a state-of-art research in our country through a selection of latest concept and application. And the selection of speakers is intended to reflect the full diversity of experiences within the community, with the proper gender, career stage, geographic position, and the research teams balance. So we will have the talk by two senior leading scientists. Professor Maurizio Prato is a worldwide recognized for his seminal work on smart nanomaterials obtained through functionalization of carbon nanostructures. whereas Anna Bernardi is a leading scientist in carbohydrate chemistry. In particular, her interest focus on the design, synthesis of sugar mimics able to interact with specific biological targets. On the other side, in the roster of speakers, we have two young early career researchers who were recently awarded by 2 ERC starting grants, in particular Dr. Flaviana Di Lorenzo coordinated a project dealing with the glycomics of gut microbiota, whereas Dr. Luca Dell'Amico project is aimed to develop new photocatalytic processes. Professor Walter Cabri is now full professor at the University of Bologna, but he spent a large part of his career in the industrial sector, so he might offer a privileged view on the importance of a close cooperation between academia and industry towards sustainable processes. And sustainability is also the research interest of Professor Renzo Luisi, who recently won a big cooperative European project, SAS Farm. Finally, Dr. Manuela Melucci is a research director at National Research Council. CNR is our biggest public research institution, and she will discuss on the importance of the organic chemistry in the achievement of sustainable development goals. In the case, goal 6, ensure clean water and sanitation to all. Please let me thank all today's speakers and enjoy this very nice afternoon together. The first, let's start. Let me welcome the first speaker, Professor Maurizio Prato, University of Trieste and CIC Biomagion, Spain. The title of his talk is Carbon Nanodots, the missing link between the molecular and the nanoscale worlds useful for photonics and catalysis. Maurizio, the stage is yours.
Thank you very much, Valeria. Can I share my screen? Can you go out?
I cannot interrupt. Okay, I found the function. Okay, sorry.
Okay, it is a real pleasure today to be here. And first of all, I want to thank the Organic Chemistry Division of the Italian Chemical Society for their kind invitation to be here with you. As you may know, we are a group of organic chemists working in nanosciences, trying to apply organic chemistry principles to nanomaterials synthesis and applications. This is a really some difficult work because, as you can imagine, the dimensions of nano objects are different from those of organic molecules. And in fact, the domain of nano objects is in the nanometer scale, and we can go to this domain starting from top, which is, however, most of the times more an attitude of physicists, whereas chemists prefer to go from bottom, starting from molecules. And but if we look at the two worlds of organic chemistry world and nanoscale world, we see a difference in size, dimension. And anyway, we can for sure say that the molecular world is completely dominated by organic chemistry because we know how to create complex structures starting from simple molecules. But also in the nanoscale world, organic chemistry can make a difference because we can modify the structure of certain nano objects, and we can make them readily available for applications. And we can also add more functionalities to these objects. So now, in what we wanted to see is whether we can find a link between the molecular world and the nanoscale world. And to do this, we started working with carbon nanodots, thanks to a recent ERC grant on this topic. And what are carbon nanodots? These are quasi-spherical nanoparticles, mainly composed of carbon, hydrogen, and oxygen, with sizes below 10 nanometers. Doping with other elements is possible. In fact, nitrogen doping is very common, but also sulfur doping can be useful. These compounds are soluble in a variety of solvents, including water. There is no big issue with toxicity, so they are relatively biocompatible. And what is very exciting about these materials is that they have absorption and especially strong fluorescence, which makes them good candidates for a number of applications. So for instance, they can act as electron donors or acceptors in their excited states. But from our point of view, they are very interesting because they have functionalizable surfaces, in particular with amino groups or carboxylic groups. So carbon nanodots are a relatively new type of material, which can be generated by, let's say, destroying objects like graphite in a top-down approach, or we can start from simple molecules, assemble them together in a bottom-up approach, and prepare these carbon nanodots. Even a lot of natural products can be used to generate carbon nanodots in what we can say is a product of incomplete combustion. When we started our work, we looked at what is available in the literature, and you can see that one of the most astonishing properties of this material is that you can change the nature of the starting materials, and you can completely modulate their emission properties. And you can see from this graph that we can go from the ultraviolet to the infrared. So just by choosing the right starting materials. In this picture, we started with a very simple approach based on the use of an alpha amino acid, which is arginine, together with a simple organic molecule, ethylene diamine. We heat this mixture with very little water in a microwave for just 3 minutes at 240 degrees, so very high temperatures. There is a sort of fast decomposition of these materials, and we get these carbon nanodots, and we have to check their nature by using, for instance, atomic force microscopy, when we can see that the size of these spherical materials is around 2 or 3 nanometers. They also have a relatively small absorption in the visible region, and they have this fluorescence, which is wavelength excitation dependent. So the emission changes while changing the excitation wavelength. In general, the quantum yield of fluorescence is not very high. In our case, we have an average of about 17% of fluorescence quantum yield. Since we are organic chemists, we wanted to check whether we can understand a little bit more about the structure of these compounds. And I must say that this is a very difficult task. No one knows the exact structure of these compounds. It is known sometimes some parts of these dots, but in general, it's very difficult. This was a work that we did using carbon-13 labeled starting materials. So for instance, in the middle spectrum, we used both reagents, arginine and ethylenediamine, completely labeled with carbon-13. And we can see that we have, of course, a lot of signals because these materials are not very well defined. We have an aliphatic part, which probably comes from these carbons and from these carbons, and an aromatic core, which is generated by condensation reactions, especially of arginine. We can purify these materials by using 2 different approaches. In one case, which is the most general used in this field, is the dialysis. So by use of dialysis with the correct filters, we can purify the dots from the from the excess starting materials and from small organic molecules. You can see here, this is the atomic force microscopy picture, and this is the distribution of the size. But we can also use size exclusion chromatography, where we can separate fractions. In this case, we separated 3 fractions with larger size, medium size, and smaller size. What is interesting, the distinction among these fractions is that their properties slightly changed, especially the quantum yield changes, because we have, you see, 7, 31, and 46% for the 3 fractions. So you can see here from this picture. And so these materials can be made really in a very easy way and can be purified again in a very easy way. And so they can be used in a number of applications. For instance, in collaboration with Professor Gouldi at the University of Erlangen, we used carbon nanodots in combination with donor moieties like porphyrins of phthalocyanins in photoinduced electron transfer reactions, so in the creation of donor acceptor hybrids. In collaboration with Francesca Danna and Renato Noto, we prepared carbon nanodot-based ionogels, and also in collaboration with Professor Gurni's University of Ioannina, we prepared clay hybrid films with carbon nanodots, so fluorescent hybrid films. But what was very interesting for us, what was a driving force for this work, is that the basic question. We have arginine and ethylenediamine, which condense somehow under these relatively drastic conditions. And so they form this kind of spherical aggregates. So the idea was, if we place some other reagents in the reaction mixtures, can we imagine incorporating this material into the dots, so changing the properties of the dots based on the nature of this material that we had? And so this was the question. And we started by just adding a new chromophore. So these body P chromophores are very well known because they have a very strong fluorescence. And so we mixed them. We mixed this compound. We placed a carboxylic group, hoping to have some condensation of this group. And so covalent linkage of this material with the other starting materials in the microwave for the formation of these carbon dots, which we expect to be green because these compounds emit in the green region. And so the atomic force microscopy tells us that the dots are still spherical. You can see them here in perspective. The size is more or less the same, around 2 or 3 nanometers. The properties change a lot with respect to the blue dots, because you can see now that the absorption spectrum is dominated by the absorption of the body P part of the assembly. And also the emission, you can see we have a very strong fluorescence in the green region induced by the presence of the body P moiety. And so we have 2 different regions. One part is the part related to the carbon dots, and one is related to the body P component. So they are relatively independent, and we can see them changing. I mean, the body P part changes also only in intensity, whereas the carbon dots, you can still see the change of wavelength according to the excitation wavelength. So now the next step was, can we generate white emitting carbon dots for photonic applications? So the white light is defined by the Commission Internationale d'Éclairage by the luminescence located in this region of the visible region. And this point of emission is usually obtained by mixing together different ratios of red, green, and blue components in order to generate white light. What we wanted to achieve was, can we start from commercial precursors and in one step obtain directly white light, white emitting nanodots? And so we decided to, following our approach, in which we add other reactants to the arginine and ethylene diamine components, we decided to use naphthalene dianhydride because in the presence of amino groups coming from ethylene diamine or from arginine, we expect this anhydride to go to the imide stage. And these naphthalene diimide are considered the most atom efficient chlorophyll mimics. They are really extraordinary fluorescing agents. Basically, all the entire visible region can be covered by changing the substituents in the core of these diamides. And as you can see here, by changing the molecular structure, we can change the fluorescence and emission, the absorption and emission properties of these materials. And this is work by Frank Würtner, who has dedicated his life to these materials, very interesting materials. So the idea was, if we take the naphthalene dianhydride and we place it in together with these two, with our reagents, we in situ generate this material, which is then mixed inside the carbon dots. And so the carbon dots which are generated by using this material, they are dominated, the absorbance is dominated by the properties of this material. And also the emission is typical of naphthalene diimide. However, we are still here. So we are still in the blue region here. And so we have not moved much towards the center of the graph. And so we decided to use another starting material, so the dibromo compound, which in the conditions, the reaction conditions, should change to monoamino bromo diimide, or maybe diamino naphthalene diimide. And so in this case, you see, this was successful in the sense that we moved towards higher wavelengths. And also the fluorescence now is towards the red region. And if we look at the graph, we see that we went a little bit too far. So we have one spot here and one spot here. And we want to go in the middle. So we decided to use them both. So if we mix them together, you see, we have this fluorescence. And so in practice, we cover the entire visible region. So we can expect a better result. And so if we use the arginine and ethylene diamine together with naphthalene dianhydride and that dibromo derivative, we can expect to end up with these materials, which we know might be helpful in moving the excitation, the luminescence properties towards what we want. And in fact, this is what we see. We see that now this is the new carbon dots. They are absorbing in the entire visible region. And as you can see from here, we are exactly at the same value we wanted to reach. So this was a very important target we reached by modeling the starting materials and reasoning the synthetic approach. Not only this, we can change the ratio between the starting materials, the reaction conditions, and we can move all the fluorescence properties on the entire visible region. So starting from this region, which we can call cool color, or the warm color in this region. So this was our effort in preparing white carbon dots. And then we wanted to see how the electrochemical properties can be changed. So the original carbon dots have two peaks in the cyclic voltammetry. We have one oxidation peak, which is probably attributed to the presence of amino groups on the surface of the carbon dots. So this is the oxidation of amino groups. And this is a reduction peak, which we don't know what it is. And so we decided that we wanted to change the electrochemical properties. And to do this, we wanted to add quinones to the reaction mixtures because quinones are very small molecules with very rich electrochemical properties. And so under the standard conditions, we can see that we modulate the structure of the quinones we add to the mixture. And we can see that the electrochemical property, you see, this is the potential versus standard conditions. And we can see that these are the original carbon dots. And you can see, for instance, here, you see from here to here, the reduction properties change a lot. The the potential is much lower, so the carbon dots can be reduced at much lower potentials with respect to the original dots. Or they can become more difficult to do oxidized. Of course, we are introducing electron attracting groups, and so we expect to have an easier reduction and a more difficult oxidation. And so these are the cyclical thermograms. You can see that now we have this peak here, and also in the oxidation stage, we can move towards more positive potentials. So what we do with this? So we use the methyl biologen, because methyl biologen has a reducing voltage, which is slightly higher than the reduction of protons to hydrogen. So this would be a photo-induced potential reduction of water to hydrogen. And so basically, what the plan was to take carbon nanodots, to excite them in the excited state, then reduce them with an external agent, and then the reduced carbon dots would reduce methyl biologen. So this is the cycle. Carbon dots are excited. We used EDTA to reduce carbon dots to reduced carbon dots, and these are able to reduce methyl biologen. And the course of this reaction can be followed by UV visible absorption, because we see the blue light, the violet light coming out, you see, in the reduction of methyl biologen. And so we can follow the reaction by using UV visible absorption. And we can see that depending on the type of carbon dots we use, we can change the kinetics of the process. We can make it faster with more stronger acceptor dots. So another issue that we wanted to study was the preparation of chiral nanodots. Chiral nanoparticles is a subject of intense interest, but usually what happens is that the people add external chiral moieties to the preformed quantum dots in this case, or graphene quantum dots here. But the properties are given to the materials by adding chiral compounds to the external part of the nanoparticles. What we wanted to do, we wanted to improve the chiral properties of the dots just during the formation step. And so, of course, these conditions are too drastic for arginine to maintain their chiral properties. In fact, the circular dichroism is completely flat. And so what we understood is that we need compounds with a more stable structure and configuration, chiral configuration, in order to keep this configuration in the final products. And so we decided to use cyclohexane diamine because the carbon-hydrogen bond is strong, and the chirality of this material can be, this carbon can be maintained at very high temperatures. And so we prepared R and S carbon dots, and you can see that the absorption properties do not change much with respect to the previous ones, but now we can see the circular dichroism of the two, let's say, enantiomeric carbon dots. Again, the size is similar to what we have seen previously. The luminescence is still there. We checked the fluorescence intensity by fluorescence detected circular dichroism. You can see that the two enantiomers behave with oppositely behavior. And we can also, in collaboration with Professor Bürgi at the University of Geneva, we did a vibrational circular dichroism. And this is the spectrum. which is, you see, very strong in this region, which is the region of the CH bending. So probably the chirality in the carbon dots comes from, as we expected, of course, from these two asymmetric carbon atoms. In collaboration with Professor Purrello at the University of Catania, we studied the chiral transfer to porphyrins. Porphyrins are achiral compounds, and since they are, in this case, tetrasulfonate, they are negatively charged, they can aggregate on the surface of the carbon dots, and we can transfer the chirality from the dots to the porphyrins. And so the aggregation behavior of the porphyrins changes with the pH. And so we started with pH 9, and we decreased the pH to 2.5, where we have aggregation of the porphyrins around the carbon dots. The absorption spectrum changes with pH, as we know, but especially the circular diachronic spectrum now shows that the Sorel band and the Q bands of the porphyrin have become now chiral. So we were able to transfer the chirality of the dots to the flat porphyrins. However, we were trying to understand whether we can generate dots with properties of circularly polarized light luminescence, CPL. And so in this case, we used this tropisomeric diamine together with citric acid and benzoquinone. And again, in the microwave, we obtained, again, these carbon dots, which have, as you can see, more or less the same size, maybe a little bit bigger because of the size of the binastal units. And now we can see that the luminescent properties are still there, and the chiral properties are still there. You can see that now we are more in the visible region, and especially in collaboration with Professor Lorenzo Di Bari, we could see that these dots have CPL, so circularly polarized luminescence. The conclusion is that we can modulate the properties of carbon nanodots in terms of molecular composition, the photoluminescence properties, electrochemical properties, chiroptical properties, and we can use carbon nanodots as catalyst or photocatalysts in organic synthesis. There is no time for me to go through this part of the talk. So this work is published, so you can see it. In practice, carbon nanodots can be used as photocatalysts for the introduction of perfluoral chelated moieties in aromatic and olefinic compounds, or can be used in aminocatalysis for the preparation, for the functionalization of bulky carbonyl groups. And the reaction is very versatile, it's very useful. And we can also use chiral carbon dots to generate chiral induction in the products. So the conclusion now is that carbon dots are easily prepared by solvothermal synthesis, starting from inexpensive and abundant precursors. The properties can be often predetermined by designing the nature of the starting materials, and careful purification is needed because sometimes the carbon dots can contain starting material or byproducts. And so, in conclusion, we see that the carbon nanodots can be considered the missing link between the molecular and nanoscale world because of their unique molecular-like behavior emerging from a synthetic protocol that we established. So this is my group, the two groups mixed in San Sebastian and in Trieste. Here are the names of the collaborators to the work that I showed you today. and I thank you for your kind attention.
Thank you, Maurizio, for the nice talk. Of course, the discussion is encouraged. Please raise your hand or interact to the question and answers chat.
Maybe Valeria, I'll ask a question if that's all right. Maurizio, excellent presentation. Thank you very much. Carbon nanodots is outside my own experience, so it's interesting to see what you've been able to do. Just from a practical point of view, in some of the last examples you showed you're able to use carbon nanodots in a sense to use them as organocatalysts. Are they filterable at the end of the reaction? So practically, how can you remove them? Is that easy or is that difficult?
Yeah, in principle, you can filter them. Yes, they are not so difficult to. You can precipitate them from the reaction mixture. So in principle, you can eliminate them very easily. You can filter them, or you can also recycle them. So in some cases, we try to recycle them, and they, for 3-4 cycles, they still work.
Very good. So Jose Virgilio Prata has a question. You're unmuted. Jose? Okay, not quite sure. Fernando Casio has also got a question, so maybe I'll pass over to Fernando. Can you ask a question, Fernando?
Yes, yes, please. Thank you very much. First of all, thanks a lot, Professor Prato, for your outstanding lecture. My question is about the emergence of chirality in these carbon dots. Is this chirality a superposition of the chiral centers included in the starting materials, or does this the configuration of the quantum dot affect the intrinsic chirality of the nanoparticle?
Oh, thank you, Fernando. The properties of the starting materials change a little bit because, for instance, they, I mean, during the course of the reaction, we have condensation reactions to form the nanodots. So the nature of the starting material changes completely. So, for instance, we expect to have, I don't know, for instance, in the case of diamines, we expect to have at least one amide formation, or, for instance, in the case of when we place, in the example I showed with binaftal diamines, we had to introduce benzoquinone as a further reactant because we wanted to form covalent linkage of the starting materials. So in a way, what we see is the chiroptical properties of the modified starting materials. I don't know how much the rest of the molecule can modify the chiroptical properties, but for sure, these are not the starting material optical properties because the nature of the compounds changes in the course of the reaction.
Okay, thank you very much.
Good. Another try with Jose Virgilio Prata. Jose?
Did you hear me? Yeah.
Yes, very faintly.
Yeah. You hear me? Yeah. Okay, never mind. I'll try to. You hear me?
Yeah. Again, maybe Jose. You can also use the chat function if you want to ask a question. You can type in the question.
Okay, I'll try that.
because it's quite hard to hear you.
Okay.
Gianfranco, I will allow you to talk now in a moment. So maybe Gianfranco, you can ask your question while Jose is typing his. Gianfranco? Gianfranco, can you ask your question?
Hello, do you hear me?
Yes.
Yes.
Okay. Good evening. Thank you, Professor Prato, for your very inspiring discussion. I just have a curiosity. I just want to ask you if it's possible, in principle, to use instead of diamines also alcohols or thiols based substituents, and if it is possible, how this change can influence the final product of carbon dots.
I can say that almost the entire molecular world has been tried in the synthesis of carbon dots. So diols or alcohols have been extensively used. Of course, the nature of the products depends on the nature of the starting materials, and so we decided to use diamines because we believe that the one amino group interacts covalently with the dots, so they are covalently attached to the dots, and the other amino groups stay outside. And in fact, by using the Ninidrin test, the Kaiser test, we can evaluate the number of amino groups we have on the surface of dots. And based on this, we can use them as catalyst, for instance, by knowing how many amino groups we have. Of course, if we change from amine to diols or alcohols, everything changes. And so, for instance, one of the most widely used substrates for making carbon dots is citric acid. In the case of citric acid, you will have carboxylic groups at the outside and not amino groups. So the properties, of course, will change a lot based on the nature of the starting materials.
Okay, thank you.
Very good. So Jose has asked his question in the chat.
Yeah, I can see it.
So TEM.
Yeah. The question is
know.
whether we can use TEM transmission electron microscopy to characterize the dots.
Yes.
And yes, the problem is the low contrast, because usually carbon is used as a substrate for transmission electron microscopy, and we have carbon on carbon. So some groups say that they can obtain dark dots, and in some cases, when we used, for instance, heavy atoms, we could see actually darker spots. And so, but in general, it's very, very difficult. So we are still at the stage where we are trying to understand whether we can establish transmission electron microscopy as a technique for characterizing carbon dots.
Okay, it's time to pass to next speaker. Many thanks, Maurizio. The next speaker is Dr. Flaviana Di Lorenzo, University of Naples, Federico II. The title of her talk is Deciphering and Exploiting the Chemical Features of Silent Lipopolysaccharide, a Gift from Gut Microbiota. Flaviana, the stage is yours.
Okay, can you hear me?
Yes.
Okay, great. So thank you for the kind introduction. And first of all, let me thank you for inviting me to this fantastic symposium. It's a pleasure and an honor for me to be part of this and to present my research activity and to share with you some recent results. So I will talk about lipopolysaccharides. Lipopolysaccharides are the main constituent of the gram-negative outer membrane that are involved in a plethora of different key biological rules that span from the integrity of the outer membrane itself till the resistance of the bacteria to environmental stressors or the presence of antibiotics. They are also involved in most, I would say, of the host bacterium interaction events, but I would say that they are mostly known for their capability to elicit an host immune response. And each of these biological rules is potently influenced by the chemical structure of these molecules. So let's give a look to these molecules. So here you can see the typical cartoon that describes how LPS is made. So you can see this is just a scheme. This is you can see that an LPS is composed of 3 different moieties. We have a glycolipid portion termed the lipid A, that is the part that is anchored to the membrane and is typically composed of a disaccharide of glucosamine bisphosphorylated and carrying acid chains, primary and secondary acid chains of diverse nature. Then we have an oligosaccharide chain that is core oligosaccharide, which is linked to a polysaccharide chain termed O antigen. This region, which is highly variable, can be absent in some cases. And in that case, we talk about lipo-oligosaccharide. This is nice. This is just a scheme, a cartoon, but the reality of these molecules can be tremendously more complex, and the chemistry of this molecule is not easy to be characterized. So, and believe me that also the tiniest modification on this chemical structure can affect the biological activities of this molecule, and in particular, the immunological functions of this molecule. But why immunological functions? Because our immune system is equipped with specific receptor, that specifically recognize the LPS. The most famous one is the TLR4-MD2 complex that specifically recognizes the lipid A portion of extracellular LPS. But now we know that we have also intracellular receptor for LPS that in some ways has access to the cytosol. We also know that the carbohydrate portion of the LPS is specifically recognized by carbohydrate binding proteins known as lectins. So overall, all the combination of these interaction events is responsible for the resulting immune response, which is typically considered as an inflammatory response that is aimed at eradicating the bacterial presence from our body. So, but you can understand how important it is to know the chemistry of these molecules, to have the key in your hands to fight, as example, against bacteria that can be dangerous for our health. But what does it mean to define the structure of this molecule? As I said, it's not an easy task, and actually there is not one procedure to do everything. I sketched here a general scheme, let's say an overview of the approaches that you can use to arrive to the structure of the LPS molecule, but actually you have to adapt the isolation procedure, and the characterization procedure step by step on the basis of the chemistry of the LPS that you have in your hands at the moment. In general, the structure of an LPS is the result of a combination of data that you can have from compositional analysis, so lipid and sugar composition analysis, NMR spectroscopy, and several mass spectrometry techniques. So as I said, not an easy task, but you can have very important information, especially if you are interested in understanding the biological rule, the function of that chemistry, or instead, the other way around, if you have a function and you want to understand the chemical structure that is responsible for their biological activity. For example, you can be interested in understanding if the structure of the LPS changes on the basis of the environmental changes, or you can be interested in defining the biosynthetic pathway of these molecules in the resistance. We know that we have a lot of bacteria that are resistant to antibiotics, and it is known that LPS has a role in this, the chemistry of LPS has a role in this. Or as in my case, you could be interested in understanding how changes of this chemistry can affect the immunological properties of this molecule. So, okay, I used this slide to show you that the LPS is widespread, and I was lucky enough to study the structure of LPS from bacteria of several natural sources, so extremophilic bacteria, environmental bacteria, or plant-associated bacteria, bacteria that can interact with insects, of course, human pathogenic bacteria, which actually were my main interest during the first year of my scientific career. And now my interest in the last few years shifted a little towards the gut microbiota world. Why gut microbiota? Because now we all know that our gut is full of bacteria that are necessary for the proper physiology of our body. And a part of this bacteria establish, an immense part of this bacteria establish harmless and beneficial relationship with us. And this, despite the continuous vigilance of the immune system. So answering questions like, which are the chemical features that render gut bacteria good and harmless to the eyes of the immune system is absolutely mandatory. And in this context, I started to think, but what is the role of LPS in the gut microbiota world? Because we don't have to forget that LPS is traditionally considered as the most potent elicitor of an inflammatory response that can be even fatal in humans in some cases. But since we know that a massive part of the gut microbiota is composed of gram-negative bacteria, means that of LPS. But if it's true that LPS is just an inflammatory molecule, how is it possible that we are not always under an inflammation status that is aimed at eradicating this bacterial presence? I do think that the answer to such an important question relies in the chemistry of LPS from gut microbiota that must be different from any other LPS structure characterized so far. So my main activity now, my main interest now is to define which are the chemical features that distinguish a commensal, let's say, so a harmless LPS from a pathogenic one. And I will show you now some recent results in this context. I'm working on several commensal and beneficial bacteria. I want to show you just a couple of examples, Bacteroides vulgatus and Archaeogenes fecalis, that are both beneficial, so necessary gram-negative present in our gut. So let's start with Bacteroides vulgatus. So my interest in this bacterium is thanks to a collaboration with Professor Frick of University of Tübingen, because she demonstrated that when she gave to mice the bacterium, so the live Bacteroides vulgatus, and these mice have an intestinal inflammation, she observed that upon this administration, there was a reduction of the symptom of this intestinal inflammation. So fantastic findings, but we were interested in the LPS. So we isolated from this bacteria the LPS, purified it, we gave her, and she repeated the experiment on mice, and she proved that the LPS alone was able to reduce the symptoms of inflammation, so just mimicking what the entire bacterium was able to do, so the restoring properties of the entire bacterium. Of course, this is fascinating because just one molecule, it is able to reduce inflammation, was fantastic. So we moved immediately to the characterization of the structure of this molecule. That, as I said before, is the result of a combination of techniques, so compositional analysis, NMR spectroscopy, and several, in this case, mass spectrometry technique. And I characterized the full structure of LPS, which is completely new and unreported. This is sketched here. And I've highlighted in colored circles some chemical peculiarities. First of all, let's look at the lipid. So here you can see that the lipid is what we normally call an hypoacylated lipid, which means that is a lipid that has less than 6 acyl chains, that is typically associated with a potent immunostimulant molecule. Here we have just 5 or 4 acyl chains. Another thing is that the typical disaccharide backbone, which is normally bisphosphorylated, in this case carries only 1 phosphate, and this phosphate is only present on the reducing glucosamine unit, which is another characteristic that suggested that this molecule can act as a weak immunostimulant molecule. Another peculiarity is the presence of acyl chain that have odd number carbon atoms, which is not so common, and branched acyl chain. Another peculiarity. As for the saccharide part, also here some uncommon features, such as the presence of the rhamnose as the first sugar directly linked to the KDO. The KDO is the marker of LPS, is the sugar that bridge the lipid to the choroligosaccharides. And this sugar is a rhamnose this time. So the rhamnose is connected to the KDO. So not an heptose as normally happens, or a mannose as sometimes happens, but still a manno-configured sugar unit, which is a branched sugar unit that carries another rare sugar to be found in the choroligosaccharides of an LPS, that is the galactofuranose, which is highly immunogenic in mammals. And then we also find fucose, which is also very important in terms of immunological properties in glycochemistry field. And then we also found exosis, and that's very simple, I would say, O antigen that is made up of a disaccharide repeating unit of rhamnose and mannose. So an overall structure that is completely new. We have this structure, we have it pure. So we move it to define the immunological properties of this molecule by using human in vitro models. And this thanks to a collaboration with Professor Bernardini of University of Rome La Sapienza and Fabrizio Chiodo of CNR in Pozzuoli. And we observed that this LPS was able to only slightly activate the release of pro-inflammatory cytokines, while it was able to selectively induce the release of interleukin-10, which is an anti-inflammatory cytokine. So a very interesting finding, a novel finding. We also observed that this LPS was acted as a very weak activator of the TLR4 signaling, while it was able to synergistically activate both the TLR4 and the TLR2 signaling. And the fact that it is able to activate the TLR2 signaling is very interesting, since the TLR2 is commonly associated with the establishment of commensalism in the gut. So it might be a way that the bacterium uses to establish a permanent and harmless relationship with the human host. So just one molecule that acts as a weak agonist, as able to release not pro-inflammatory cytokine but anti-inflammatory cytokines, and that is able to activate both the TLR4 and the TLR2. And then we also went at a molecular level, trying to define the molecular details of the binding of this LPS to the TLR4 MD2 complex. And this thanks to a collaboration with Professor Martin Santamaria. She proved that there were some thanks to, of course, molecular dynamic studies, she proved that there were some predictive binding poses that were halfway between the agonistic conformation and the, let's say, less agonistic conformation. So she proposed that the coexistence of these two putative binding poses may be responsible of what we observed on human cells, that is a very weak capability of elicit an inflammatory response. Of course, we now are trying to dissect the immunological signaling of this molecule, and also we are interested in understanding the role of the carbohydrate part by studying the interaction with specific lectins and how these affect the signaling of these molecules. Okay, let's move to the second bacterium, Alcaligenes fecalis. This is another very interesting bacterium because this bacterium is able to colonize the Peyer's patches. Peyer's patches are a gut lymphoid tissues that where the IgA production occur. So it was very interesting for us because Alcaligenes fecalis is a gram-negative post-SLPS that, as we say, is considered as immunostimulant. So how is possible that an LPS is there, so a bacterium with its LPS is there in a tissue that is so related to the immune system? So once again, we characterized the structure of this molecule, and once again, a new structure with some chemical peculiarities. In this case, you can see these are lost, so there is not the O antigen. And starting again from the lipidae, also in this case, we found hypoacylated lipidae, let's say mainly hypoacylated lipidae, penta and tetracylated were the main species. We, in this case, detected some hexacylated lipidae species, but these hexacylated lipidae species have a 3+3 symmetry, which is commonly of the acid chain, which is commonly associated with lower immunostimulant activity. Another peculiarity is the presence of, as a secondary acid chain of hydroxylated acid chain, which is not so common. It seems to be related to the establishment of a permanent relationship in a tissue for a bacterium. For the saccharide part, here you can see that the first sugar directly into the KDO is heptose, as most commonly found. But I want to point the attention on the fact that this choroligosaccharide had a massive presence of N-acetylglucosamine and N-acetylgalactosamine, which is not so common to be found in the core of an LPS. So, again, new structure, but I wanted to show you this example to highlight another reason why I'm interested in the LPS from gut microbiota, because I think that we can take this molecule as an inspiration model to conceive, to synthesize derivatives. And this is what we did in collaboration with Professor Fukase, that synthesized the hexacylated, pentacylated, and tetracylated lipid species of Alcaligenes fecalis lipidae, and he tested this molecule, these synthetic compounds, for their immunological properties, and he observed that the penta and tetracylated lipidae were unable to activate the TLR4 mediated response, while the hexacylated form was able to do it, but at a less extent compared to the Escherichia coli LPS, which is the most commonly used LPS to induce an inflammatory response. So he demonstrated that the part that is responsible for a weak agonistic activity of this LPS was the minor component that it was of the lipidae that was hexacylated. But coming back to the LPS, they also investigated if this LPS was involved in some cases in the IgA production, because as we said, this bacterium colonizes Peyer's patches, and Peyer's patches are responsible for the production of IgA. And what they saw was that the LPS was able to enhance the EGA production. So a very nice result. And they also evaluated the adjuvanticity properties of this LPS to induce antigen-specific immune responses. So just to tell you that one molecule that is a weak agonist, a promoter of IgA production, and that has adjuvant properties, and in fact, they proposed this molecule as a effective nasal adjuvant to augment IgA antibody and Th17 cell responses. Okay, so what's next? Of course, as I said before, we are now interested in understanding at a molecular level the role of the core oligosaccharide, the O antigen, the lipid, so each portion of the LPS in the immunomodulatory properties that we observed, and also in the establishment of commensalism. Of course, I'm interested in also define the signaling pathways of this molecule, which is expected to be different from what was known so far about LPS. Of course, we will continue to synthesize derivatives to dissect the immunological properties of these molecules, and why not dreaming in any application of these molecules for clinical practice. Okay, so I hope that I've convinced you that it is time to break the ancient dogma of LPS as just a toxic molecule, especially in the light of the new rule of the LPS in the gut microbiota, and to consider this molecule as an inspiration model to prepare derivatives that can prevent or reverse inflammatory processes. And let me say that I also think that the LPS from gut microbiota, so these weak agonistic molecules, are necessary for our body, and are necessary at a specific time of our life and at specific sites of our body to prime, educate, and train both the systemic and local immune system. And with these thoughts in my mind, last year I started to think even bigger and wider, and to conceive the bugging LPS proposal that I submitted for an ERC as an ERC starting grant proposal that was accepted for funding on last December. And so I want to spend just 2 words about this new adventure for me. This is a structure to function study for which I set 3 specific goals that will help me to reach the main goal of the bug. Let's use an informatic terminology, the obsolete concept of LPS as just a toxic molecule. So I will want to prove that this molecule can be even beneficial for us. So 3 specific goals. The first goal, I called it the chemical language. Sorry, is not okay. The chemical language, yes, because I do think that, as I say, that in the gut, the LPS speaks a different chemical language. I will provide information about this chemistry and immunology related to this chemistry. Then I will translate all this information in the production of chemical tools, that is analog, synthetic derivatives, derivative substructures that are inspired by LPS from gut microbiota, that will help me to understand which are the key epitopes responsible for a specific immunological behavior. But I said that I want to prove that LPS from gut microbiota can be even beneficial for us. And to do this, I will use preclinical tools. And those preclinical tools, I decided to use a specific type of staminal cells differentiated in brain cells to reproduce in vitro a neuroinflammation process, and to see if any of these molecules, natural or synthetic, is able to rescue from the pathological phenotype. So why the brain? Because we all know that the gut is connected to the brain, that the gut microbiota potently influence our behavior, our social behavior, and in general, the development of our brain and of our brain immune system. So I'm really interested in the understanding the role of and the impact of the LPS from gut microbiota in this gut-brain axis. And of course, there is a, let's say, a personal reason that was the provocative choice of the debugging LPS proposal. That is because today pathogenic LPS, so Escherichia coli LPS or Salmonella LPS, are considered the most used tools to induce inflammation in vitro and in vivo models to study neuroinflammatory processes. So my question is, what happens when we use this weak agonistic LPS in this kind of models? So please stay tuned, because I really hope to come back to you with the interesting results on this topic. Okay, just to conclude, I want to thank all the group in Naples of University of Napoli, Federico II, the Department of Chemical Sciences, my mentors, of course, Tony Molinaro and Alba Silipo, and please all the colleague and PhD student, but please I want to thank in particular the Marie Curie PhD student Molly Peter that is in charge of the gut microbiota, of the LPS from gut microbiota characterization, and her work and their enthusiastic work is priceless for me. And of course, all the external collaboration and all of you for your kind attention.
Thanks, Flaviana. I think that the time is over, so we have the time just for one question. Please raise your hands.
So Valeria, Peter Bellotti has a question.
Good afternoon, can you hear me?
Yes.
So I have a question regarding the epitope of immunogenic LPS. So you said like these are basically unknown, but do you have an idea whether it's the sugar part or the lipid chain part that is playing the biggest role in the non-immunogenic response of these LPS?
Okay, good question, good point. Thanks for the question. Okay, we know already that most of these gut bacteria, the commensal one, have what we call this hypoacylated lipidae. So already the fact that they are hypoacylated at the level of the lipidae means that they are unable to induce a potent inflammatory response, which is just one thing. We also think that there are some sugar on the carbohydrate part that are specifically recognized by what we call tolerogenic lectins. So lectins that recognize this glycoepitope and so they can elicit tolerogenic mechanisms, so not harmful signaling pathways.
And Valeria, I think there's one further question from Alessandro Casnati.
Hello, can you hear me?
Yes.
Yes.
Okay, great. Nice talk, Flaviana. Thank you very much for your talk. More than a question is a curiosity. You are proposing with this new project to have some new LPS which are beneficial for our gut and so, and it's very interesting, I guess, and it's very difficult target also because you showed that just changing very small groups, you immediately end up with different biological properties. So can you anticipate something more about your approach you are going to follow?
Okay, you mean to study how these changes affect the immunological properties? How we will study this?
Yes.
Okay. Yes. There are several approaches. So for a reason of time, I didn't tell everything. Of course, I will study this first in the typical in vitro cellular models, human cellular models. So like Hex cells transfected with specific receptor, or macrophages. I will follow the interleukin profile to understand if that molecule is immunostimulant or not, which kind of interleukin it simulates the release. Then I will move to a more, let's say, gut-like model. I will use Caco cells. I will use also co-culture to understand, to mimic in vitro the gut inflammation and to see if any of this molecule is able to restore the normal condition. And then I will, everything will be translated in the brain system. So first I will study microglial cells, so the immune cells of the brain, and then these more complex brain cellular models. So I will reproduce in vitro, let's say, the brain, of course, in cellular models. So I mean microglia and neurons and so on, and see what happens.
Thank you very much.
You're welcome. Thanks for your question.
So we welcome the next speaker, Professor Walter Cabri, University of Bologna, and Innovation and Development Fresenius Kabi. The title of his talk is Green Pharmaceutical Chemistry, an Inescapable Need.
Thank you, Valeria, and thanks to the Organic Chemistry Division for the opportunity of today. I hope that everything is fine. Okay, you see here three logo. Why? Because we are working in several areas, from drug discovery, mainly on peptide and peptidomimetic for protein-protein interaction. Then we have a lot of projects as a green innovation, and we are also part of the Center for Chemical Catalysis here at the University of Bologna. Now let's see if it's working. Okay, this is the team here at the university. I share the leadership with Alessandro Formelli, and then we have several PhD students here, and we have Tommaso, Chiara, and Dario that are working mainly on catalysis. Tommaso is in John Hartley team in Berkeley now, and all the rest of the team is mainly involved in projects related to green chemistry and mainly on peptide chemistry. These are two teams.
Walter,
yeah.
let's just pause there for a second. There's a lot of background noise when you're speaking. I don't know if you're using a microphone or what.
I'm not using a microphone.
Yeah.
It's really awful.
It's quite hard to hear you. I don't know if anyone else hears that. Yeah, the audio is bad. Sorry now for interrupting you.
No problem. Let's see if I'm able to rapidly connect the. Sorry.
No, it's okay.
Now you can hear me?
No, there's still, as you talk, there's a bit of a rattle.
Only a second. Only a second. Now? Not yet.
Yeah, it's as you speak.
Only a second. It should come. Okay. Only a second. Can you hear me now or not?
We can hear you, but each time you speak, there's a little bit of a rattle at the back.
Second.
Sorry now.
Okay. Now things are fine.
I'm afraid it's still not great. I don't know if I've got some other mails in the chat to say that the audio is bad. It wasn't a problem when you spoke earlier.
You should be connected now, no?
Yeah, that's a bit better. That's better now.
Working now?
That's better, yes.
Yeah.
Okay, sorry for the inconvenience. Now, let's see how can I, okay. So let's go back from the beginning. Okay, this is the team at the university. I'm leading that with Alessandro Tormelli, and we have all the PhD students that are involved in our project, and this is the innovation and development in Italy of Fresenius Kabi. Actually, this is only 50% of the team. And then we have in NZPEP our colleagues in the Netherlands that are working on the development of new enzymes. The collaboration between industry and the academia has been always very critical. In all my career, once I was on the industry side, or now that I'm also in the academic side. Mainly because in the academia, we can get better education target, real work targets. And on the other side, the access for the industries for new technology, material, and of course, driving all this for excellence. This is coming from Phil Barron slides. Of course, nowadays, the work is a lot influenced by the sustainability, and the thematic and the topic of green chemistry is becoming really central. These are some of the cross-coupling reactions. As you can see in the palladium catalyzed reaction, there has been a lot of delay between discovery and implementation at the industrial level. These are the patents you can see here. Mainly Suzuki Miyaura, but also Ekasar, Sonogashira, and Mizuki Eka are among the most subject to patent. Some time ago, I had to work on, you see, this is a Suzuki, sorry, is an Ekasar Sonogashira reaction in order to get this intermediate. This is an intermediate for erlotinib that is a non-small cell lung cancer drug, oncology drug. The process that we have defined was based on a Sonogashira coupling, so palladium and copper. And actually, it was very good, high purity, high yield, no color issue. The issue where on the other side, the Sonogashira is a Sonogashira protocol, so you need copper and palladium. PMF is an absolutely toxic solvent, very dangerous for the liver, and in addition, we need extra purification for the palladium removal. We decided to look back at that reaction and understand if we can do something better. And now if you look at these three, this green tree is our interpretation of the 12th principle when we talk about cross-coupling reaction, especially palladium cross-coupling reaction. I wanted to highlight only the prevention of waste, and the prevention of waste is measured nowadays using PMI, the process mass intensity, and that's the way to drive the development of chemical processes. The solvents and the auxiliary are critical and better if these are coming from a biogenic and sustainable source, and also energy. 6 is really critical. Why? Because, and I think the lesson that we are teaching today is that, of course, energy is very important. We need to avoid extreme reaction both on the high side and also on the lower side, if possible. And then, of course, talking about catalysis, you need processes with an high ton and high TOF. Looking back at the Ekasar-Sonogashira reaction, we saw that, okay, there was some work carried out using green solvent, but not much. So we decided to enter on that. We found that, especially using, in this case, the Sonogashira condition, so palladium and copper, you can run the reaction under very mild condition, 25-30 degrees in a few minutes to half an hour, not more than that, using 2% catalyst. And the good is that HEP, that is the hydroxyethyl pyrrolidine, is a green biogenic solvent, so potentially is largely available from natural sources. We decided also to go further and deeper to understand why it's so less toxic with respect to DMF or NMP. And at the end, the reason is very simple. It's because the metabolic pathway is totally different. Instead of generating formaldehyde, you are generating a sort of derivative of glycine. Now, we have used also a specific base that historically I've used because it's very high. The apparent PK is around 12, 13. And in addition, because this base has a very high green score, also compare with, I don't know, potassium phosphate. The other thing that you have to fix in some way to have a green and sustainable process, not only the solvent, the base that you are using, but also the fact that you're dealing with palladium. I like palladium, is very potent and very reactive as a metal, but it's rare, you see here, and it's dramatically expensive. It is only because of the real cost of the extraction and purification of palladium, but of course, because it's subject to a lot of speculation. Anyway, if you have to use it, you have to use it in very low quantity, and in addition, you need to recycle as much as possible. This is the mechanism. Why I show the mechanism? I show the mechanism because the stoichiometry is not always perfect when we talk about the Eka-Sonogashiya reaction. In several cases, you need to have an excess of the alkyne. Why that? Well, this is the EuChemS protocol, so palladium and copper. Clearly, the transmetallation process is less efficient than the Sonogashiya protocol, where you have the copper. And recently, Cosmic came up with this different proposed mechanism, where you have two palladium cycles, one that is activating, of course, the aryl halide, and the second one that is activated and transfer the alkyne. Actually, this should lead to a side product that is the homo coupling, but in reality, this is not present. What is present, on the contrary, is the anion. So you have the competition between the palladium zero oxidative addition on the aryl halide, or the oxidative addition of the palladium zero with the alkyne that generate the anion. That's the reason why we think that, at least in our condition, the EuChemS original model developed also by EuChemS a few years ago is still the most interesting one. So pi coordination and then switch to the sigma complex. The way to avoid, of course, the formation of the anion is simply to have a slow addition of the anion. Slow addition means in 1 hour, 2 hours, not more than that. And then you get complete reaction, having only a slight excess of the alkyne in your process. So having said that, we have extended reaction on all these products. The excess of the alkyne was very limited. What we observe is that during the workup, and we carried out the reaction in water, the extraction was carried out with toluene, hexane, or azobutyl acetate. I mean, you can go also with very green solvent. What's happened is that you do not have contamination of the product with the metal, and that's interesting. So the metal was, I mean, not contamination of the HEP into the organic solvent. So the idea was then to use HEP water as a solvent, and instead of using triphenylphosphine as ligand, using sulfonate phosphine. Very simple idea, very simple thing. No, not very new. But what was new is that when you run the reaction in HEP water, the catalyst remain in the HEP water, you extract your product, and then you can recycle the catalyst. If you start with 2%, you can recycle the catalyst up to 15 times, and you go a very, very low mole percent of the palladium. Now go back to the erlotinib synthesis. So you have the EuChemS protocol, you generate the intermediate. Of course, you need the acetylene to be protected on one side, otherwise you have the double coupling. Then simply you extract the product, you add a little bit of sodium hydroxide, you reflux, you deprotect, you concentrate and wash the toluene solution with water. Then you add isopropanol, intermediate 5, and you got the product. And this can be run in a telescope process without isolating any intermediate. If you use the Buchwald ligand, what is happening is that you can also recycle and so decrease the impact of the catalyst and also the app. And so we started with 0.2 molar percent of palladium, can do 3 recycles, you go very low with the palladium, you have good top, but in addition, you have a process mass intensity of 15 for the entire process. If you recycle the solvent and the base mainly, what you have is only 5. That comprises also all the chemicals that are required to recycle the exhausted palladium catalyst. Simply, you add a little bit of formiate, you reduce to palladium 0, you add a little bit of charcoal, you filter, and then you can recycle the catalyst. So in this way, we are able to get a very efficient process, not very long, the process itself. And in addition, we can recover 100% of the palladium that actually, as you saw before, is very, very expensive. Now I'm going to change completely the subject and move into another area where the impact of try to green the process can have a really great outcome. This has been a challenge for a lot of groups worldwide. This is the typical solid phase peptide synthesis. And as you can see, the first part, you make the linear peptide, and then you have to purify. In the upstream process, the main solvent they're using is again dimethylformamide, and that's not nice. And what's happened is that, again, the first point you have to solve here is to change the solvent. Then you can think about the rest, the base, the coupling reagent, and the PMI. These are the groups that work in that area. Has been a challenge for several groups. We are talking about academia and also industry. Somewhere we have also Eli Lilly, not only Novartis or Novo Nordisk. And these are the 3 papers that we have recently published on the subject. The review that we have made in this area came up in Green Chemistry early 2022 and became an article and is now free for download. So anybody can go there and download for free this review. That summarizes all the effort of the last 5 years on greening SPPS. SPPS is very important because it's the technology that allows to incorporate inside of the linear sequence of the peptide a natural amino acid and what is necessary to have, of course, a more druggable product. Nowadays, you can have products that are orally available, very long half-life, semaglutide that is the best in class for anti-diabetes and anti-obesity, has an half-life of 1 week, is oral available, and is a blockbuster of 10 billion euros. So these are the work that we have performed. Solvents on the ACS sustainable, we have worked on N-octylpyrrolinol that is, by the way, very safe, and we have also worked on alternative and safer base. Is one of the best base in terms of green score. This is the linear octreotide. It's a very sequential reaction, standard SPPS, and we are able to get the final purity of the product that is 100% identical to DMF, and we can also use automated synthesis by simply decreasing the viscosity of NOP, that is one of the main issue there, simply by adding dimethyl carbonate, keeping the system green and having good selectivity and very easy recovery of the solvent, decreasing the impact and the PMI. We are now working on other coupling reagents, but what I want to discuss now is the combination of this feature with the enzymatic coupling that you can perform with peptides. If you have a long peptide and up to 40 amino acids is still considered also from a regulatory standpoint a peptide, what is happening is that even if you have high efficiency in the coupling, the protection sequence, etc., you end up with a consistent amount of impurity that then you have to purify. So what's happened is that you can, for example, stop the reaction at 20 and maybe divide it into the product and make the coupling. And in this way, able to increase the purity of the final product, the concentration of the final peptide, and make all the purification process very easy. You can do that, for example, with an ligase. That is a unique technology developed by EnzyPep, that is part of the Fresenius Kabi team, in collaboration, in this case, also with the Groningen University. The reaction, of course, goes under very high condition, very high catalytic efficiency. The key point is that this reaction is in water and do not require any protective group on the rest of the amino acid. So very simple. The nice and interesting thing, and this is the example of ozorotide that was approved in 2021 for achondroplasia, is that, of course, subtilisin, these are all enzymes coming from the subtilisin, is an hydrolytic enzyme. And then you can take the ozorotide and you can subject to hydrolysis with subtilisin. Of course, not a normal subtilisin, a subtilisin that belongs to our library. So you understand where you can have, you know, hydrolysis in the sequence. In this case, you can have hydrolysis between 16 and 17, 21, 22, etc. Why we perform this reaction? Because you understand where you can use the coupling. Because the same enzyme moving from the serine inside the active site to the corresponding cysteine, the enzyme became an enzyme ready for coupling reaction, not hydrolysis. So very simple, very fast. In this way, we can get immediately the site where we can perform the coupling and then move back. I describe this very easy. It's not really like that because most of the time you have then to design, you have to optimize the active site in order to have very high yield, very high conversion, very high selectivity for your specific substrate. But these are the pocket of the subtilisin on the acid side and on the amino side, and working on this pocket allow us to increase consistently the productivity, the selectivity, and getting good results. And actually yesterday, Silvia Rizzo, that was a postdoc that was shared between University of Bologna and EnzyPep, got her degree, and she was working on this project on ozorotide. This is my final slide. I hope that the beginning was not so dramatic. The problem with the noise, I hope that now is fine. This is nice because it's going back to 1537. It's the beginning of the well in Orvieto, and it said, well, nature cannot deliver what we provided by human work. And that's what we are doing. Nature can provide very nice peptide, for example, but if you want to have a drug, then I think human work is necessary to get it working. We have worked with the University of Parma for the metabolic study, University of Ferrara, the group of Professor Cavazzini, for the purification of peptides, and of course, Fresenius Kabi, that means also EnzyPep, and Enrico Faggi, that is a company that provides us all the catalysts and work with us on the recovery of the palladium catalyst that I described in the first part of the process. And this is the final slide, and this is the Navile, that is the campus, the new campus that we are going to have that we have in Bologna. We are going to move in September. It's very nice, and we welcome you if you want to come shortly. Thank you.
Thanks, Walter, for your nice talk and for being perfectly in time. Now the discussion is open. Please ask your questions.
So Valeria, Gianluigi Albano wishes to
Yes. Do you hear me? Yes.
Yes.
Okay. So thank you very much for your very nice presentation. Very inspiring. I have just some curiosity on the first part of the presentation concerning the Sonogashira coupling. The first one is related to the purification of the compound. The isolation was very interesting, but after the isolation, all the compounds were then purified by column chromatography, or the purity was very high in order to no further purification. And second, very big curiosity, why in your work did not try to use, for example, heterogeneous palladium catalyst, since they could be used more times? In the literature, there is the possibility of a higher reusability of this kind of catalyst. Thank you.
Can you hear me without noise or with noise?
Unfortunately, the noise is back.
Let's see if it's now. It's working, no?
It's better, but yeah.
Only a second, let's see if it's working. Sorry, but it was unexpected. Now, still?
Noise.
And it was working so well, Walter. It was working so well.
I don't know why. I take out the earplug and then I don't know. I don't know if it is because there is low battery. I try to be very fast, or is it a disaster, total disaster?
No, I mean, we can, it's possible to make out what you're saying. So please do try.
Okay. So, of course, to characterize the product, we carried out and to have a perfect yield, we carried out the chromatography. But the product does not require any chromatography. In fact, in the where we have a fully optimized process, you do not have to do anything. You do not have any contamination of palladium, ligand, or solvents, or whatever. You go directly behind. So we have always products that do not require any excess of reagents and so on. It's absolutely fine. For concern, that was the first question. For concern, the second question, I'm from the industry, and the industry now also, I think worldwide, are suffering for two things. The first one, the cost of the solvent is not going back to the pre-COVID era. There are solvents that are still 3-4 times more expensive than what it was in 2000, at the beginning of 2029. And the second point is really energy. So if you move from homogeneous to heterogeneous catalysis, and you look at the temperature that you normally use, you're always talking about very high temperature and generally very high load. In order, for example, you have two trends in green chemistry. The first one is palladium. You go very low with the amount of palladium, recycle as much as possible, and then recover a lot. Lipschutz is going to work in that area. By the way, we are superior to Lipschutz in terms of PMI and similar in terms of quantity of palladium. And then there are people that work with flow chemistry, where, for example, you can use, in that case, heterogeneous catalysis. You try to perform the reaction much faster, but then you have to go 130, 140 degrees. So at the end of the day, it's a balance. I don't like reaction. When I see higher than 100 degrees, I don't read the paper. For me, is a nonsense, because I have this, industrial approach, and that's what we need to be efficient. We are working at 80, 90 degrees with arylchloride. It's not in some preparation of the paper, but with arylchloride you can work easily at that temperature, and the reaction are very fast. In 4 hours, everything is done with 0.2% molar percent palladium, or even less. Okay.
Okay, thank you very much.
Very good. So Valeria, there's another question from Lorenzo Guazzelli, who was a former postdoc actually in Dublin with Stefan Offerson. So he's now asking the question. Hi, Lorenzo.
Hi, Pat. Yes, I have a question related to the cost of these solvents, especially of this HEP solvent. Is it cheap enough? And considering that is a kind of new one, is it already available in ton scale for, if companies are interested in using it?
Okay, so the cost now is consistently higher respect to DMF. It's 6 times higher. DMF is around €1 and something. This is around €6 and something. But that's because of the fact that you are producing still million of tons of DMF. Hemp has the good thing that if you solve the problem, if our colleagues from the biology solve the problem of succinic acid production, is a very straightforward process in that case, and not using the normal chemical approach they use now for the pyrrolidone. There is a nice paper that I presented on the green chemistry from another authors. And the driving force there is that hemp is an intermediate for vinyl pyrrolidone, so also for the polymers. So they can come from a natural source, from lignin. So in the future, if you want to work on sustainability, that's the way it can work. Now, having said that, in the process that I show, the 5-to-1 is mainly a little bit of water, 2-3% of hemp, and only a few percent of other chemicals. But the hemp is almost all recycled. So even in that case, the impact from a cost point of view is limited.
Okay, thanks, Emilio.
Valeria, I can see two further questions. One from Lehosh Kovacs.
Yes, that's correct. Okay, my question is that this solvent that you try to apply, this 2-hydroxypyrrolate, is quite expensive. So what is your proposal? I shortly scanned your recent review in green chemistry, and you listed quite a lot of solvents according to different principles. So what is your perspective about this particular solvent?
As I said, I think in the academia, we have to show what are the potentials of certain chemicals. Then, of course, you have to solve some issues that are always related to the one of biogenic solvent. The fermentation is the key, not the chemistry to get the hydroxypyrrolate. And it's not my purpose.
No, sto scrivendo
I think in the future, as a perspective, that is the route where we have to go, because circular economy is based on this. So I think that's the way.
Okay, thank you.
Great. And Virginie Vidal, my French colleague on the Division of Organic Chemistry. Hi, Virginie.
Hi, Walter. Can you hear me?
Yes. It's a pleasure.
Yes, how are you?
Fine, thank you.
Do you remember Montreal? So we were in postdoc.
We share the hood, no?
Yeah, we share the hood, exactly. So first, a very nice lecture and very instructive lecture in terms of green chemistry. So my question is related to the Ek-Kassar-Sonogashira coupling to access the erlotinib synthesis. So you make very smart recycles with 0.2 mol% of palladium. That means 0.06% of palladium to access the anti-cancer agent. But just a simple question, how do you remove totally the palladium?
Luckily, the presence of the sulfonate phosphine will remain in the water 100%. So what we're going to get after pre-recycle is that the conversion is likely less than, is going close to 90%. And since we do not want to have the contamination of the raw, of the starting material, we stop the reaction. But in reality, it's not zero. So all the catalysts remain in the water. And that's what is nice about it. So you can recycle, and there is no contamination of the product because, this is a drug, and so we have very low limit in terms of elemental impurity.
Okay, so it's a really green process. Thank you. Hope to see you.
Hope to see you too.
Bye-bye.
Thanks, Walter. Let's go to the next speaker, Professor Anna Bernardi, University of Milano. The title of her talk is Interfering with the Sugar Code Design and the Synthesis of Glycomimetics. Anna, the stage is yours. Anna, we cannot hear you. Please switch on the audio.
Sorry, I turned on the screen share too early and I forgot the audio. Thank you very much. Can you see it now? Can you hear me okay?
Yes.
Okay, so thank you, Valeria.
Anna, if you could maybe just make the screen bigger by just going to the slideshow.
Yeah, I did, but has not worked.
Oh.
Okay. So sorry about it.
Well, you can make it maybe bigger with the 100%. You can go with the thing down on the bottom right. You can just make the thing a little bit bigger.
This is actually as big as it gets. On my screen is large, is full screen. Don't you see it this way?
No, we see all your other slides down the left-hand side as well.
That shouldn't be how. Don't understand why.
Anna, bisogna che condividi l'intero schermo e non solo...
È condiviso tutto, Maurizio. I'll just do it again, but that's how it works for me, at least.
Perfect.
Okay, sorry, don't know. Well, thank you very much, Valeria, for the introduction, and thanks to everybody for the opportunity to be here today. It's been very interesting and very exciting so far. So I'm going to share with you some of the thoughts and the results that we have developed in this program of ours about the design and synthesis of glycomimetics. The reason why we want to develop something that is really not going normally. Sorry about it, but my arrows are not working. It was working perfectly when we tried at the beginning. Don't know what's going on. I'm stuck.
Okay.
I'm trying with all the possible,
We can see your cursor.
Okay, I'll do it this way. It's a bit clumsy, but it seems to be working. So reason why we are developing glycomimetics in the first place. I think you've seen some beautiful examples of how carbohydrates and carbohydrate-protein interactions are becoming the target of a lot of interesting studies that are working towards the development of probes and all the way to drugs to interfere with these sugar-protein interactions, because they are at the core of a number of physiological and pathological phenomena in the biological world that we would like to get hold of. And in order to do that, glycomimetics can be a lot more effective than natural oligosaccharides, because very much like peptidomimetics, they can be made more drug-like than the natural ligands. They can also be made significantly easier to synthesize than complex oligosaccharides. And they can be, with time and luck, more active and even more selective than natural sugars. The kind of challenges that we are facing are many, but most are really related to the fact that sugar-protein interactions are intrinsically low affinity. Typically, as we always say, they are in the millimolar to the micromolar range. And this has a lot to do with both the structure of sugars, that they are very happily solvated in water and don't see very many reasons to interact with proteins, and also with the typical binding sites of sugar binding proteins, which are collectively called lectins. They tend to have binding sites which are very large and shallow and solvent exposed. And this, in the end, means that the affinity is largely dependent on solvation-dissolution. So what you get is that you have an interaction that is computationally extremely hard to predict. And having these binding sites so large and shallow also means that you don't have the typical excluded volumes that make it easier to use docking tools and stuff like that. So you have something that is complicated to begin with. And on top of that, we also have that many lectins have overlapping specificity, meaning that they recognize the same monosaccharide. So there's a question of selectivity, which has rarely been addressed. The way these problems have been tackled by us and other groups has been multifaceted. So many groups, including us, started from what I call design based on the structure of the ligand. And from there, so we try to design structural mimics of oligosaccharides. From there, we moved to a design that I like to call the sugar anchor, and I'm going to show you an example later in the talk. And both us and essentially everybody else in the field have exploited multivalency as a way of increasing affinity through avidity. While doing so, the principles that we try to use are here in the blue boxes. We wanted to avoid glycosidic bonds to make things easier to make and more difficult to hydrolyze. We wanted to increase lipophilicity to increase the bioavailability of these systems, but also to increase the affinity for lectins through a specific lipophilic interaction on the surfaces. And throughout all this, we wanted to keep it simple. Keeping it simple helps you to do screening programs where you can screen a relatively high number of structures. So it will help you in your discovery process. And also is essential to exploit multivalency in a meaningful way. The examples that I'm going to show you today are all related to what has been the most developed project in this program of us that is connected to a specific lectin called DC-SINE, which is a membrane receptor of dendritic cells. DC-SINE is a lectin, is a tetramer. It has 4 carbohydrate recognition domains that all contain one functional calcium ion. That is where the sugar connects. The tetramer is held together by this coil-coil neck tetramer that goes through the membrane of dendritic cell. The physiological role of DC-SINE, like other pattern recognition receptors in dendritic cells, is to recognize pathogen with the goal of internalize them, degrade them, and present them to T cells for the start of the adaptive immune response, and therefore of an effective immune response to these pathogens. However, it was discovered that many pathogens, and the first one that was discovered in this sense was HIV, actually take advantage of this receptor. And so they are recognized, but rather than being degraded, they actually use the receptor to be carried along in the host body. So the final effect is that rather than attacking this pathogen, DC-SINE is concentrating them at the surface of dendritic cells. So it's creating a very effective high concentration of a very high viral load that helps the infection process rather than fighting it. So the ability of disrupting this interaction would help fighting the infection by reducing the viral load and by allowing other therapies or our own immune system to effectively fight the virus. This is sort of the principle of anti-adhesive therapies that are actively sought for, both in this case and in other situations that are similar to this. So the specificity of DC-SINE is for mannose oligosaccharides and also some fucosylated oligosaccharides. But the one we concentrated on is this high mannose oligosaccharide, which is the most expressed glycan at the surface of HIV. And the binding epitope of this for the lectin is actually this 1212 or 1216 D and 3 saccharides that are effectively recognized, although at relatively at low affinity, which is, as I said, typical. And our initial step into this field was the design and the synthesis of these pseudo-D and pseudo-3 oligomannosites, where essentially we replaced the reducing end mannose with a cyclohexane diol, which is conformationally locked and enantiomerically pure, so that it represents structurally a 1,2 disubstituted mannose. This molecule in particular, the pseudo-D saccharide, has exactly the same affinity as the native disaccharide for dicycine, but fulfills many of the criteria that I was discussing before. It has this unnatural aglycon, which increases the lipophilicity and also increases the enzymatic stability of the structure. And it's already equipped for conjugation to other aglycons or to the multivalent scaffolds that we want to use. It also has a much simpler synthesis than the disaccharide itself. The simplest we have so far, I'm going to show it to you very briefly. It's actually for the equivalent thiodisaccharide or pseudo-disaccharide, which can be made in a one-pot reaction starting from the mannothiolacetate here. Using DMF, I'm sorry, Walter, you remove with diethylamine, selectively remove the acetyl group here, and the thiolate attacks this epoxide, and owing to the properties of this epoxide and to the stereoelectronic requirements for its opening that are strictly transdiaxial, you can only get trans attack here or trans attack there relatively to the carboxy group, which gives you exactly the same molecule. So you end up with a very efficient process to make this. You can also do it with the aziridine that can be either Bock protected or already conjugated to glycans of your interest. And in this way, you actually have a better possibility of linking this through a more stable bond while maintaining exactly the same affinity and increasing the stability through the thiol group here, the hydrolytic stability of the system. So, but let's go back to the interaction with dicycine. This is the pseudodisaccharide. It's a crystal structure that was obtained in the lab of Frank Fieschi in Grenoble. We have been collaborating with him over more years that I care to mention on this project. It's been great. So the green thing here is the calcium ion. The sugar moiety connects to it. The aglycon additionally interacts with the protein. And this is the very large and flat and shallow binding site of dicycine that I was talking about. At the beginning, rather than trying to design in that region, which would be exceedingly difficult, we decided to modify the molecule in the easiest possible way. So we developed derivatives where it was chemically possible. And we ended up with a series of amides, among which we selected this ligand that has a slightly improved activity over our starting disaccharide due to more extensive interaction with the surface of the protein. But, and that was totally, serendipitous, has selectivity over langerin. Langerin is a related lectin, similar to dicycine, but happens to be protective in HIV infection. So if you want to to use these molecules in the context of fighting HIV, certainly you don't want to interfere with Langering. So in this case, just screening and discovery was the approach. From there, we try to use something more rational, and we paired with Sonsoles Martin Santamaria. She performed a fragment-based screen on the structure of the complex that you have just seen. And among other things, she identified an ammonium binding site which sits in this region of the molecule at the foot of this phenylalanine 313, which happens to be closer to the position of the oxygen 2 of the mannose ring. So we developed a number of ways that we can connect an amino group essentially to this position here. Turns out that the most effective was also the easiest one to make through this triazole. And this molecule actually increases the affinity of the system in a very significant way. We're going down to 50 micromolar, which is essentially one of the most active, if not the most active, monovalent ligand for this design, which is available at the moment. So this is the kind of structures that we like to call sugar-anchored glycomimetics, in the sense that you can still barely see the structure of a sugar in this molecule, but the part of the sugar that we maintain is the one that is actually anchoring your molecule to the lectin, sort of exploiting the ability that lectins have evolved over millions of years to recognize these residues, and then adds additional interactions that come from the aglycon or from modifications that you can make even on the sugar part. And these interactions with secondary sites are providing you with higher affinity and also with increased selectivity for this kind of molecules. This is totally selective against Langering. We have not really checked it yet, but also unlikely to be recognized as a mannose by any hydrolytic enzyme because of the modification of the sugar ring. So we think this is an interesting way of approaching the problem. And even more interesting possibly is that fragment screening into the structure of the sugar-lectin complex appear to be viable. It's one of the few viable computational techniques that we have been able to adopt in this project so far. The other thing that we could do with these molecules was to present them in multivalent fashion. We go back now to the structure of this MAN30. Remember, it was the first Langering selective ones that we found by serendipity. Its activity is about 300 micromolar against Langering, and we made them into multivalent systems, dendrimers here, in collaboration with a group of Javier Rojo in Sevilla. These molecules already are able to inhibit this transinfection by HIV. That is the process I was describing before. This design is carrying the whole virus to its final cellular target. For HIV is T cells. And this is what you measure in this kind of experiments. The affinity is still only in the low micromolar range for these kind of things. But the real breakthrough occurred for this kind of structures. There are still dendrimers with a rigid core at the center. There is a rod shape, so you see it's very rigid and really a small rod. Here now in the same kind of experiments with HIV, you get into the nanomolar range. And the same occurs in the case of SARS-CoV-2. Frank Fieschi and Rafael Delgado have shown, indeed, that this design interacts with the spike of SARS-CoV-2 through the glycan shield of the virus. And although also in this case, we don't really have a direct infection of the dendritic cell, the trans-infection process occurs here with ACE2 expressing cells. Now, this process can be blocked by polyman 26, as we like to call this rod center system. This polyman 26 is actually effective at blocking the interaction of this design with the spike protein, and also is effective at blocking the transinfection process in concentration that are in the high nanomolar range in these cellular studies. So it seems to be a very efficient design. And I like to spend the last minutes of my talk discussing the design of this molecule. The initial design came from the observation that this PM19, the smaller dendrimer that I showed you at the beginning, remember, 5 micromolar, it's really too short to actually bridge across 2 binding sites on this design. The distance between the 2 calcium ion is about 4 nanometers, and this molecule cannot go across. So the only mechanism that we are exploiting here to improve the affinity of this from the monovalent to the polyvalent system is essentially statistical rebinding, the ability, or if you want, the increased effective concentration once the first binding event has occurred. The design, as I said, came from the idea that we could chop this in half, and we could insert in this a rigid system to separate the 2 halves of the dendrimer. Of course, we tried with a number of possibilities, and this rod here is the most effective one. It's rather large, so you have more than 23 Armstrong going from nitrogen to nitrogen, and then the 3-valent system provides you with density, local density on the 2 binding sites. I can show you the effect, the data of how these elements have an effect on the interaction of these molecules with this design through these direct interaction experiments that were performed by SPR in the lab of Frank Fieschi. So once you connect MAM30 to this rod system, you already have 1 order of magnitude increase, which is due to the interaction of the rod with the surface of the protein. But as soon as you make it divalent, you end up with a structure that is just long enough to, in fact, chelate, effectively chelate to calcium binding sites. And this provides you with another order of magnitude, in fact, is almost 20 times, increase in affinity, going down to the low micromolar level with just a divalent system. But if you also add the possibility of including statistical rebinding here, a little bit more flexibility, and a little bit higher concentration of the ligand, now you get 2 orders of magnitude at this point, going down to the, let's say, 12 nanomolar, which is very consistent with what we are seeing in the cellular studies. So we can say, and this is one of the first time that in the literature this was actually observed, that chelation and statistical rebinding are amplifying one another in this system in a way that becomes very effective, although the valency of the system has not increased very much. We did not have to build a huge polymer to get to this kind of effect. It's just a hexavalent system, but it contains all the right elements that allow you to effectively exploit chelation and statistical rebinding at the same time. Just for a final thought, this design is a tetramer, so we try to exploit this by engaging all 4 binding sites simultaneously using this cross-shaped kind of systems. Of course, the length of the diagonal is larger than 4 nanometer. It's between 5 and 6. There are 2 diagonals that are not the same, 5 or 6 nanometers. The extended length of this system of Polyman 26 is over 6 nanometers. So it is possible in principle that this cross system can reach all 4 at the same time. It was not a great success in terms of affinity. We are here essentially at the limit of the assay, but for this system, which bears a lower affinity spearhead, we can see there's basically no increase in affinity, but there is, and that's serendipitous again, an increase in selectivity. So the selectivity against langerin, which was already very significant for Polyman 26, is further increased. And we think that it has to do with the fact that this design is a tetramer, but langerin is a trimer. And so the complementarity of the scaffold and the complementarity in topology between scaffold and lectin appears to be contributing to the selectivity of the system. And I think I did not overdo my time, so I can just highlight some conclusions. Some of things that were successful were design of glycomimetics based on the structure of the natural oligosaccharides involved, and that can evolve through structure simplification and or addition of binding elements that you can select rationally using fragment screening. Evolves to this concept of using sugar as an anchor and decorating it in viable ways. The selectivity, we got it by serendipity. You can get it by differential design, which I didn't have the time to show you, just using features, different features in the same regions of different lectins. The most important, though, multivalency, that so far we've been unable to do without, because in the micromolar range, we haven't managed with monovalent system to go below the micromolar range yet. So multivalent constructs, we can strive to use all the possible mechanisms of multivalency, and you can see that they can amplify one another in very significant ways, and it's in this sense easier to go down in affinity. This is it now, really. So I can thank all the people that you have seen in the slides and the people that you have not seen in the slides, the group in Milano and the various collaborations that we had in these projects and other projects. Thank you very much for your attention.
Thanks, Anna, for your nice talk. The discussion is open.
There's a question coming through from Sebastian Goin.
Oh, bonjour, Sebastian.
Bonjour, Anna. Can you hear me?
Yes.
Okay, also thank you for your very nice talk. You show that when you block DC sign, you perturb the interaction between the COVID virus and DC sign with your multi-monocytes. Does it mean that the virus is covered by accessible monocytes?
Yep. The viral spike you can see here. This is actually a picture from Frank's lab. You see that, and these are cryo-EM that were obtained also in Frank's lab. You can detect DC sign, which is the elongated system here, and this is the spike, right? So the spike has, depending on the variant, between 23, 27, more or less, glycosylation sites, and many of them are highly monosylated. So yes, DC sign can recognize the spike in various positions, as you see from the EM, even places where there are two DC signs attached to the same spike protein. Yeah.
Okay, very interesting. I have another quick question, if I can.
Yes. For me, yes.
Yeah, it's about the rigid linker. You have some acetylenic bonds.
Yes.
And we know that acetylenic bonds can react, for example, with short group from proteins. Did you see some kind of covalent grafting to short from proteins, or the?
We haven't really looked for it. So we haven't seen it, but we haven't looked for it. So maybe we should.
Okay. Okay, thank you again for your talk.
Thank you.
Other questions?
I don't see any other. Maybe everyone is in need of a cup of coffee. I don't know.
I sure am.
Fact, say us what is the return time. And we are a little bit late.
Yeah.
According to schedule, please.
Well, I would suggest maybe if we could even have a break for 8 minutes and start again at 15:45.
Okay.
Sorry, 16:45 for you.
Okay.
Okay.
Okay, so 8 minutes. Enjoy.
Okay, thank you.
Enjoy.
Bye.
Thank you.
And our president, Organic Chemistry Division of the Italian Chemical Society, Professor Valeria Dauria. Okay, I will show today some recent results from our lab. We are involved in the use of a chemical technology for developing a sustainable synthetic methodology. In particular, we are involved in the field of flow chemistry and flow technology. Well, the concept behind the flow technology is very simple. So what you need is just teeny tubes or micro devices that are fed with a solution of reactant, A and B, for example. Then they are mixed, and you continuously, under steady state condition, you can collect the product C. What is important is the mixing zone and the mixing unit of your flow device, because sometimes the design is very important to control some very fast reactions. Two important features of the flow systems: fast heat transfer and fast mass transfer. What this means? That you have perfect control of a chemical parameter or process parameter, and this results in a better chemical process that usually are considered more safe and environmentally friendly, and more reproducible and more selective compared to batch processing. Another point is that when you speak about a microreactor, you can think that it's possible to produce only a small amount of a compound, but this is not true, because it's possible to produce from kilograms to tons, just putting several reactors in parallel, or adjusting the design of your reactor. And now people have developed a mobile production unit that can produce on-site and on-demand chemicals or product from for fine chemical or medicinal chemistry purpose. Another point is that this kind of mobile production unit based on flow technology, they have a small footprint, and for sure in the future they will substitute the traditional batch reactor. Another point is that, for example, exothermic reactions, they are slow and wasteful in batch and are better controlled using the flow technology. Also, when you use flow technology, you have the possibility to design reagent, for example, and also to explore a forbidden chemistry. And this is an important point because there are a lot of constraints when you use the batch processing in the production of fine chemicals or medicinal chemistry product. Another point that is very important now is that micro reactor technology and flow technology can be combined with digital technology, artificial intelligence, and also robotics. And it's possible to create what is called closed loop experimentation cycle. So you have, you can design your experiment, you can use the flow technology for execute your experiment, then you can check the output with the process analytical technology, and then with the algorithm, you can optimize your process. This is something real that already work. For example, if you see this work from Clive Jensen at MIT, he was able to use a published reaction, a commercially available compound, and artificial intelligence in order to select a route for the synthesis of a pharmaceutical targets, and then flow technology for executing the reaction, and then robotics for the implementation. As you can see in the video, I hope it's possible to see it. You can see the connections, and this is a plug-and-play system that can be used not only for the production of pharmaceuticals or in general for chemicals, but also for discovering the reaction and exploring a new chemical space. In this context, I would like to introduce today the SUS Pharma project that has been funded by the European Union under the framework of the Horizon Europe program. And this was funded for 6.9 million euro. And in this consortium, there are leading institutions and leaders in the field of single atom catalysis, flow chemistry, digital technology, sustainable chemistry. And I have the honor to be the coordinator. And also 4 pharmaceutical companies are involved. The aim of the SUS Pharma project is to use artificial intelligence in order to plan a more sustainable synthetic process, use flow technology as an enabling technology, and robotics all together in order to develop new synthetic strategy and to develop also green pharmaceuticals. The project will start in September, so I hope to have the possibility in the future to talk about the result of this journey. Okay, another fascinating aspect of the flow technology is related to the possibility to control very fast reactions and the possibility to control very or highly reactive intermediate. Professor Yoshida introduced the concept of flash chemistry, defined as a field where it's possible to control extremely fast reaction in a highly controlled manner. So we can distinguish 2 different types of fast reactions. So we have a very fast reaction with a rate below 1 second. And in this case, if you use the flow technology, the design of your microreactor should be adapted to the process that you want to study or you want to exploit for synthesis. Other kind of fast reaction are reaction with the rate in the record in the range of a second up to a few minutes. And in this case, they are kinetically controlled, and heat exchange is very important. So not mixing in this case is a crucial parameter, but the possibility to have an accurate control, for example, of the temperature of your flow system. Here you can see some of the technology that can be employed to control fast reactions. And we have a conventional T-mixer or T-plate, for example, flow plate, for example, or a system designed with a static mixer, or integrated systems that allow the control of reaction that have rate from 1 second up to sub-milliseconds. Why is this important? Because if you want to exploit a reaction between a precursor in red and a reaction partner in green, and you have a reactant that trigger the reaction of a precursor in order to generate an intermediate that will react with the reaction partner in order to get your product, this reaction scheme can be productive only if you use in batch condition the internal quenching protocol, and the reactivity of your precursor should be higher than the reactivity of the partner with respect to the reactant. The other combination, they are unproductive in batch, because in one case, if you use the internal quenching and your partner is more reactive than your precursor, then in that case, you will not generate the useful intermediate for your reaction. Or if you use the external quenching protocol, so you can generate the right intermediate, but if the lifetime is too short, so it decomposes before reaction with the reaction partner, you will not get your product. So flow technology can help in order to handle this kind of problems in synthesis. And I will show today some example, and I will show how in the case of fluorinated carbenoid, for example, or in the case of lithiated heterocycle and sulfur functional group, the flow technology outperformed batch chemistry, allowed to discover new chemical space, and allow to handle highly reactive and unstable intermediates easily. So let's start with carbenoids. Carbenoids are very useful reactive species that can be employed for synthesis. They can be generated by starting from monohalo dihalomethane using an organolithium by lithium halogen exchange, or by using a lithium amide by deprotonation. Anyway, they are very delicate and fleeting intermediate, because they have a problem due to the high thermal instability. And in particular, they undergo alpha elimination or Wurtz type coupling. So they should be used under cryogenic conditions, usually below -78 degrees, and using the internal quenching conditions. So they are suitable candidates to be investigated using the flow technology. Some recent report from Kappe groups demonstrated that using the flow technology based on Lonza flow plate and air-filled technology is possible to manipulate bromomethyl lithium at -20 degrees under internal quenching conditions. But chloromethyl magnesium chloride, so magnesium carbenoids, can be manipulated under external quenching conditions using this technology, and the trapping can be done after the generation and after 1 second, soon after the generation. So we contributed to the field, investigated the reactivity of chloroiodomethyl lithiums that can be generated by reacting chloroiodomethane with LDA. And we found that using the internal quenching protocol at low temperature and using benzophenone as electrophile provide an interesting alpha chloroaldehyde in 55% yield. However, if we want to improve the yield and maybe use the external quenching protocol, we found that quenching after 1 minute provide only 18% yield of the decided product. The mechanism of this reaction is very simple. So we have a nucleophilic addition to the carbonyl group, then intramolecular cyclization, and then the chloroepoxide undergoes to 1,2 manual type epoxyl aldehyde isomerization to provide the corresponding alpha chloroaldehyde. So using deuterium as electrophile, we wanted to estimate the lifetime of this fleeting species. And what we found was that at -78 degrees, after 1 minute, more than 60% of the species decomposes, so it's lost. And this is why the external trapping is not possible to observe in high yield under batch conditions. So we investigated the process using a flow setup, and we wanted to estimate the lifetime of the lithiated intermediate. And by using just 16 experiments and interpolation of the data, we were able to build the optimization map. And you can see that the product can be obtained in 94% yields, working at -20 degrees and using a residence time of 330 milliseconds. However, if you go down with the temperature, for example, at -78 degrees, you know, the lithiated species survive for 9.4 seconds, and you can get 87% yield of your product. However, under optimized conditions, it's possible to see how the predicted optimization works, and you can see that the scope is quite wide, so the yield has a medium from good to very good. But if you compare between the flow protocol and the batch protocol, you can see that with the batch protocol, the yields are always very low. Another point is that the chemoselectivity of the process is very interesting because, for example, if you have bromine on the aromatic ring, you don't see any competitive exchange reaction. So it's highly chemoselective. And in some cases, we found that we needed to induce the isomerization under acidic conditions. So we collected the outlet of the flow reactor in a flask containing amber list 15. And in this case, we promoted very fast isomerization. And as you can see here, the reaction works very well. And you can get several different kinds of alpha chloroaldehyde that are not easy to make. And in this case, the reaction with alpha chloroacetophenone, for example, is not possible to perform in batch. We did not succeed in isolating the corresponding product, even working at -78 degrees. Another important topic is the fluorination of organic molecules, and in particular, very recently, Hartwig and Karagasov showcased the importance of fluoromethylation and monofluoroalkylation strategies in two very interesting reviews published on Angewandte. And we work in this field, and we developed in collaboration with Vittorio Pace the first nucleophilic fluoromethylation strategy based on the use of fluoroiodomethane that is not an ozone depleting agent, and using an internal quenching protocol. And we were able to generate fluoromethyl lithiums that, however, under external quenching condition, even after 1 second, did not provide the desired product. So this is an example of a very, very unstable fluorocarbenoids. However, we transferred the process in flow and we use it as electrophile, an electrophile that gave very low yield when the reaction was run under internal quenching condition. It's because this kind of reaction partner is not completely compatible with the lithiation conditions. We did this work in collaboration with Aichiro Nagaki, and we found that using 13 milliseconds as residence time here in R1 is possible at -6 degrees, is possible to obtain in the product, the desired product, in 84% yield. You can see here the scope of the reaction. They are just some selected example. You can see more example in the paper, and you can see that the reaction outperformed the running flow, outperformed the batch protocol in all cases. Another important things is that if you have, for example, special electrophile, so a system where you can recognize two different electrophilic functionality, so you have a problem with the chemoselectivity of the reaction. Well, if you run the reaction in flow, you have very high chemoselectivity, and if you compare, for example, this example in the middle, you can see that the reaction in batch is provide low yield, but also is not chemoselective. So this is an important point when you want to control, the chemoselectivity of your reaction. Using the flow protocol, will give you several advantages. We investigated also a different fluorocarbenoid, in particular fluoroiodomethyllithium, that was generated by reacting LDA and fluoroiodomethane in a flow reactor at -40 degrees. And in this case, we found that with 82 milliseconds was possible to obtain 99% of the stannilated product using tin chloride as a suitable electrophile. You can see here some example. And in all cases, we found very good yield of the trapping product. And using a carbonate compound, we were able to isolate the corresponding fluoroepoxide. We decided to demonstrate the usefulness of the flow protocol in a one-flow multi-step process. In particular, we generate the fluoroiodomethyllithium, we trap with the tin chloride, we obtain the corresponding stannilated compound, and then we subjected the fluoroiodostannyl compound to another deprotonation in order to get this quite interesting carbenoids. You have a quaternary carbon with the two different halogen with different electronegativity and two different metals. Anyway, if you want to trap this lithiated intermediate, you should use 8 milliseconds, so very short residence time at -40 degrees. However, the trapping with the carbonyl was successful, and we obtained for the first time this kind of unknown epoxide bearing the fluorine and the tin group. Using imine as electrophile provides the corresponding fluorostannilated acidity. We tried to perform this reaction in batch, but was quite impossible. We never observed the corresponding product. So we went further. We decided to investigate also the reaction of fluoroalkyl lithium, because this is not explored, but also they are expected to be more unstable compared to the fluoromethyllithiums. So we generated the corresponding starting material. We prepared the corresponding starting material, and we performed the reaction under internal quenching conditions. And we found that quenching the corresponding fluoroalkyl lithium after 1 second did not provide any reaction product. But if you run the reaction under internal quenching condition, so you are able to obtain the product even with good yields. However, we investigated this reaction under flow conditions, and again we built the map, and we found that working with a residence time of 206 milliseconds at -78 in this case, is possible to observe the trapping product in 80% yield. And we also verified that we have a transition between internal quenching regime to external quenching regime, changing the residence time. Because at short residence time, we have still good yield, but we realized that we were working in internal quenching conditions. So the rate of exchange was too low if you work at a low residence time. This is the scope of the reaction. Now we are still exploring this chemistry with more charming electrophiles, but you can see that this reaction run in flow outperformed the reaction that can be run in a batch condition under internal quenching condition at lower temperature. Well, we wanted also to investigate the reactivity of lithiated strained heterocycle, and we were inspired by this work by Agarwal that reported for the first time the lithiation of this aza bicyclobutane that was used in the Matheson chemistry in order to assess to substituted azethidines. So in this reaction, in this synthetic protocol, you can see that it's possible to use phenylithium, secbutylithium, and terbutylithium. So different organolithiums, and you have to use low temperature, -78, for each steps. So in collaboration with Oliver Kappe, we decided to investigate in flow this useful process. And in particular, we were looking for a greener and more sustainable process that could be run at higher temperature and that could avoid the use of phenylithium or thermotylithium. And also, we explored the possibility to use other electrophiles. Here is the optimized process. We optimized the process and we found that second butyllithium or even normal butyllithium can be used as the single lithiating agent. And the reaction can be run at 0 degree using 14 minutes for all the steps, lithiation, cyclization, and partial lithiation. And at 25 degrees, it's possible to trap effectively the lithiated azabicyclobutanes. And you can see here the scope is quite wide, high chemoselectivity. And in one case, we were able even to isolate good crystals suitable for X-ray crystallographic analysis in order to confirm the structure of these interesting compounds. In other protocol, we were using normal butyllithium. And we scaled up the reaction and we ran the reaction for more than 4 hours. So collecting the, the product with a productivity of more than 9 millimole per hour. We ran also a reaction, the same reaction reported by Agarwal in batch that that gave the corresponding azetidine, borylated azetidine in 54% yields, and we found that the reaction works in flow, and we obtained the same product in one flow with 84% yield. Continuing in this field, Agarwal reported recently the application of this strategy for the synthesis of spiroazetidine that are useful from a medchem point of view. And also we wanted to expand the scope of this protocol in flow, and we wanted to explore a new chemical space. In particular, we were interested in unknown motifs that by chemical manipulation can provide very rare or unknown functionalized azetidines. So we modified our original protocol, and in particular, we used chloroalkyl ketone as electrophile, chloromethyl, chloropropyl, and chlorethyl ketones. And we found that the use of chloromethyl ketone provides directly the corresponding azabicyclobutane linked to an oxirane, but with the, and you can see here the scope of the reaction. And the reaction is highly diaster selective also, and highly chemoselective. So we have only 1-2 addition, and they are quite interesting structural motifs, because you can see the coexistence of a strained azabicyclobutane and a strained oxirane. In the case of chloropropyl and chlorobutyl derivative, we found that the corresponding chlorohydrin can be isolated with the use of the flow protocol, but reaction with under basic condition provide quantitative transformation into the corresponding oxetane or tetrahydrofurane, bringing the azabicyclobutane. And this kind of scaffold is completely unknown. Further, we investigated different electrophile, in particular alpha chloroimines that were obtained from alpha chloroaldehyde that we prepared in flow, as reported before. And we found that using this kind of electrophilic acceptor, we were able to obtain the corresponding azabicyclobutane linked to the aziridine. So we have, again, two highly strained heterocycle in this coexisting in the same structure, and this is also an unknown motifs. While the use of nitron provides the corresponding hydroxylamine that under acidic condition quantitatively is converted into another unknown motifs, a spiral compound bringing the azetidine ring and the oxazetidine ring. And we are exploring further the scope of this kind of reactions. Speaking about the azetidine, we realized that azetidine lithated in position 3 are not known, and in particular, they can be easily made starting from this 3-iodoazetidine. And we found that it's quite difficult to generate and trap these lithated azetidines in batch, because we have a protolysis, and we are dealing with the secondary alkyl lithium, and that's reported by Noshel. They are quite reactive, and they undergo protolysis and should be generated under very low temperature in order to slow down the protolysis reactions that we observed in batch. But if we run the reaction in flow, and we use also cyclopentyl methyl ether as a green solvent, using 82 milliseconds as residence time at -50, we are able to control the protolysis reaction, and the product B, that is the side products, can be obtained in less than 5%, while the desired product is obtained in 80% yield. Under this condition, optimized condition, this is the scope of the reaction, and you can see that the protocol works very well, even with the reaction, in the acetylation reaction, in polylation reaction with aldehyde, with imines. So it's a quite robust protocol. We use the same starting material also for performing the synthesis of the azetine and the lithiation in one step. So we use LDA in this case, and we optimize it again in flow. We found that in this case, you need a 9.4 seconds in order to promote elimination and then lithiation in the flow system. And you can run the process at 0 degree. In this case, the product was obtained in 89% yield, and the reaction works quite well. So this is the first example of functionalization of this kind of azetine in flow, using also a green solvent and LDA as a lithating agent. The reaction works very well even for silylation with estrone, for example, with imines. So it's compatible with alkylated chain and also with unsaturation. So they are quite interesting compounds. In fact, if we collect the corresponding carbon-2 functionalized azetine in batch in the presence of oxalic acid and water, we promote a quantitative transformation into very interesting beta-aminoketones. You can see here the process work with the several different kinds of carbon-2 functionalized azetines, even, for example, acylsilane that are very difficult to make. The reaction works well, and the mechanism is quite simple. So we have protonation, and then the synthesis, the formation of this iminium ion, addition of water, and then rearrangement to the corresponding open chain. The reaction works also with estrone, and this is quite an interesting method to introduce the beta-aminoketone or beta-aminocarbonyl unit into a scaffold, molecular scaffold. Okay, in this last part of my talk, I would like to show some recent results in the field of sulfur functional group, in particular as an analog of traditional sulfur groups, such as sulfoxamine, sulfonimidate, sulfonimidamide. They are other analog of sulfur 6 functional groups, and they are very useful in medicinal chemistry because they show they give better result if compared to the corresponding sulfonamide, sulfonate, or sulfones. Similarly, for sulfur 4 functional groups, sulfinimidate ester or sulfinamidines, they are hardly accessible molecules, but still very useful for medchem. And we were interested in the synthesis of this kind of sulfur 4 functional group, in particular NH2-free sulfinamidines, and effective methodology are not present into the literature. And we thought that probably using a sulfanilamide and a acrylamide, we would have give the possibility to synthesize, to prepare the sulfur diimide that by reacting with an organometallic reagent and quenching with water should provide the corresponding sulfinamidine. Sorry. So the mechanism is this one. So we have the nucleophilic addition to the sulfur of the acrylamide, then the elimination of lithium silanolate, sorry, and then nucleophilic addition to this electrophilic sulfur will provide this intermediate that will decompose in water to give the corresponding sulfinamidine. So this is the optimized processing flow. The reaction can be run at 20 degrees, so at room temperature. The first residence time for the preparation of this sulfur diimide need to be run at 15 seconds. And then the reaction with the organometallic reagent can occur at 20 degrees and with a residence time of 23 seconds. So in less than 1 minute, you get quenching with water, you get your product. You can see that the yields are very, very high, and this protocol is difficult to run under batch condition. And in 2 cases, we reported the first crystal structures of this kind of functional groups. We also decided to investigate the chemoselectivity of the reaction. So we mixed the sulfanilamines with an arylylide, and we use a normal butyllithium in order to generate the corresponding arylithium that should have react to provide this kind of compound. And as you can see in the scope, the reaction is highly chemoselective. So you can see here the process that occur in the first flow reactor. So we have the synthesis, the formation of the sulfur diimide, and then you have a selective lithiation by exchange of the halogen with the lithium promoted by normal butyllithium, but we did not find this side product. So there is a perfect control of chemoselectivity using this flow approach. Okay, I hope to be on time. Give me just 1 minute to thank the people that worked to the project that I reported today. I would like to thank Professor De Gennaro and Marco Colello, Leo Mussi for contributing to the flow chemistry development in the lab, Michael Andresini for working in the field of sulfur functional group, Mauro Spennacchio for working in the field of fluoroalkylation. All my students and also my international friends that contribute to implement several of the project and the results that I show today. I would like to thank also Dompe for financial support, the European community, and all of you for your kind attention.
Thank you, Renzo, for your nice, a little bit futuristic talk in the first part. The discussion is now open.
So Valeria, we have a question from Anna Lenarda. Anna? Oh. She certainly had raised a hand.
Okay, I have a question in the chat.
Oh yeah, great. Maybe I can read it out because not everyone can see the chat perhaps. So it's from Graham Barker. Thanks for a great talk. I noticed for the acetidine and halocarbonide work, you were using syringe pumps. I was wondering if you know what pressures you were working at and if there is a possibility of using piston or peristaltic pumps.
Hi, Graham, and thanks for the question. And well, I don't know exactly the pressure I'm working with, but as you certainly know, that kind of syringe pumps, they are high pressure syringe pumps because the back pressure is very high when you work with the stainless steel microreactor. And peristaltic pumps, piston pumps, I don't think will be suitable unless they don't work at least at more than 50 bars. But peristaltic are not suitable because they don't have a constant flow.
There is a space for another question. We are little in delay, so please ask your question. Otherwise, we will pass to the next speaker.
Maybe Renzo, a very quick question. Excellent talk. I really enjoyed it. It's certainly expanding chemical space, and you're looking at some very strange molecules. You're getting different types of units onto some biologically active technique motifs. Have you done any biological evaluation of some of the compounds that you've prepared?
This is a very good question. Thanks, Pat, for asking. But at the moment, we don't have information about that because they are very recent results, but we want to evaluate. In vitro, we will do that.
Okay.
Thank you.
No, thanks, Renzo.
Okay. Let's welcome the next speaker, Dr. Manuela Melucci, National Research Council. The title of the talk is Advanced Nanomaterials to Ensure Clean Water and Sanitation to All. Manuela, the stage is yours.
Yes, good afternoon to everybody. Can you see my screen?
Yes.
Can you hear me? So I can start. So first of all, let me thank the division for inviting me. It's a real pleasure for me to be speaking here today. I will give in these 20 minutes an overview on our activities of the last 5 years, let's say, on the development of materials based mainly on graphene for water purification. My title comes from the title of one of the Sustainable Development Goals identified by the United Nations. As you know, we are facing very huge problems with water. First, coming from the climate change, so water scarcity, and also from water pollution. And so the topic is quite urgent. And in the last few years, we were almost overwhelmed by the huge interest from several industry and agency dealing with water treatment. The problem is also, it is not involving only the developing countries where people suffer from lack of water, but also our countries. And I'm sorry to say that we are so good in producing chemicals, synthesizing chemicals, but then these chemicals sometimes get dangerous for the environment and for ourselves. And that's the problem of Europe and developing countries. So you know that we introduce into the market thousands of new compounds every year. They, of course, are important for us. I'm talking about drugs or agricultural products, pesticides, industrial products. But unfortunately, most of them in different way reach our water resources. And today they are called emerging contaminants. And unfortunately, they are not completely removed by the conventional treating technologies. And I will give you a brief overview on this. And this is why, to face this issue, all the regulation agency are trying to update the legislation. And this is the case of Europe. Last year, the European Commission introduces introduced this new drinking water directive with new restriction parameters, approaches for water safety and control. And this is pushing the academia and industry for searching new approaches for removing these contaminants and provide safe water. This is just an example on two classes of compounds that are matter of great concern. Perfluorinated alkyl substances known as forever chemicals. They are used in thousands of products that we use, including clothes, and they are particularly persistent and mobile. So they've been found in Italy. It's a huge problem in the northern regions, even in tap water. And also pharmaceuticals are matter of concern. Just think to antibiotics. They have been found in a lot of water resources in rivers. This is a very recent study showing how in 50% of our rivers, some molecules that we use every day for different reasons have been found. So basically today we have two main technologies for removing these contaminants, these molecules from water, that are based on membrane filtration and adsorption of activated carbon. So these two technologies are used both at domestic level, so point of use devices, and also at municipal scale. The problem, the most famous case of membrane filtration is reverse osmosis. We can use it also in our home. The problem is that membrane filtration requires a lot of energy for working, because you have to push water through these membranes. On the other hand, activated carbon is cheap, but it is activated and regenerated at very high temperature. So you produce a lot of carbon dioxide for burning the vegetal sources of this carbon. And there are also ethical issues that I always would like to highlight, because these vegetal sources are collected in, and produced and treated in developing countries without any safety protection for the operators. And this movie comes from one of the major producers of activated carbon from their website. So if the last global big invention in water treatment was reverse osmosis, a few years ago, at the beginning of this century, the potential of nanomaterials start arising, because firstly, nanoparticles of metal, nanosilver, were introduced for water disinfection and now are used also in commercial devices. But then it came clear that nanomaterials can be the future for let's say, integrating several functionalities in the same materials and to integrate the currently available technologies. And this is because we can tune the properties of these materials by chemical modification. So we can combine, for example, specific sorption or photodegradation, or we can bind catalysts and induce degradation. And so since the beginning of the century, a lot of articles have highlighted the potential of nanomaterials. And among them, graphene is playing a role because of, first, because of the wide chemical versatility. It's carbon, so it can be prepared by graphite. You see here a nanosheet of graphene oxide. You have several functionalities on the surface of the sheet that can be used as binding sites. And also, beside the mechanical strength and processability and all the technical issues, graphene, one of the key advantages that it is nowadays commercially available at large scale. The prices are lowering and they are expected to be further lower because of the increasing demand from the market. And they are approaching the cost is acceptable for water purification market within the next few years. But beyond this motivation, one of the key factor of graphene is the possibility of chemical tailoring. So here you see that some examples from our lab in the last few years of chemical modification of graphene oxide. Graphene oxide is an oxidized form of graphene. You have epoxide ring groups on the surface or carbonylic, carboxylic groups. So basically you can treat them with conventional organic chemistry reactions. So for example, here we graft these molecules by silylation, or you can exploit carbon-nitrogen coupling, or you can exploit the epoxide ring opening reactions, or brushes of PMMA by ATRP polymerization. In this case, we bound a phospholipid. We developed these materials for other application also, not only water purification, because graphene is also conductive. So for example, we are working for exploiting specific recognition with transduction of these recognition events for sensing purposes. So in our lab, what we are doing is to try to tailor graphene absorption properties by chemical modification. We study, of course, these materials in terms of structure, morphology, selectivity towards several contaminants of concern. And then we also process these 2D nanomaterials in 3D composites by using polymeric scaffolds such as natural polymers, ketose and alginate. And the idea is to exploit these materials as sorbents. And today I will mainly talk about polysulfone and derivatives membranes embedding graphene or derivatives or coatings with graphene layers. So here, just to give you a shot of the absorption properties of graphene and reduced graphene oxide in comparison to activated carbon, which is this industrial benchmark we have for sorption purpose. So you see here we select a number of molecules representing several classes of emerging contaminants. Some of them are included in the European watch list of priority contaminants. And basically we found that in most cases the kinetic of the sorption of graphene and reduced graphene oxide was faster than that of activated carbons. And this is very important for point of use application. I will come back to this application later on, because we have to work with filters and materials connected to our tap. This is our main application. And we prove this fast kinetic on other contaminants, including also long chain PFAS molecules. The working mechanisms of absorption of these materials mainly rely on the interactions between the surface groups and the molecules. Of course, it depends on the molecules you are considering, the target contaminants. Here we demonstrated in collaboration with Professor Talizin of Umea University that the number of defects in terms of holes on the surface and carboxylic and carbonyl groups is directly related with the absorption properties of these nanosheets toward 2 dyes and 2 antibiotics that we use as reference molecules. And we tailor these defects by using different oxidation conditions. So basically, oxidation of graphite to get graphene oxide is done by harsh conditions, by harsh oxidant in acidic conditions. And so basically interactions with these functional groups and pi staking also as the main working mechanisms for absorption. But also another mechanisms can be intercalation through shape complementarity between the molecules that are generally in the size of these holes that enable the intercalation of these species in between graphene layers. And this is very important when we coat membranes and polymers, porous polymers with graphene coatings, because the molecules can intercalate in between the layers of overlapped sheets, and this allow to get nanofiltration performance that is quite important to remove small molecules. And we demonstrated this by filtering through these membranes coated with graphene, this molecule and other molecules. This is a ofloxacin antibiotic. And we demonstrate by X-ray diffraction and modeling that there is a change in this spacing, so in the distance between the nanosheets upon the intercalation of the floxacin molecules that was confirmed also by molecular modeling. But we can also play with these interactions, surface interactions with contaminants, and this is what we are trying to do. In this case, for example, you see that if we change the surface charge of graphene oxide that is negative because of the carboxylic groups and all the oxygen-based groups, to positive values, we demonstrated that we can remove not only cations, heavy metal cations as lead, but also arsenic anions. Arsenic, it is a very important contaminant in groundwater. I'm not showing the analytical data because it's quite boring, I know, for organic chemists, but basically we could remove 90% of arsenic anions in water by using this PEI modification. Polyethylenamine is a polycation in these conditions, working conditions. We graphed this polymer, branched polymer by epoxide ring opening, and we developed this a method for purifying this graphene, modified graphene oxide. And this is now the standard method we use in our lab to purify the substituted graphene oxide systems. Because when you deal with chemical modification with graphene oxide, the only way to purify the target compounds is centrifugation. So repeated centrifugation and washing steps that is quite tedious and time demanding, and it is not a standard way from lab to lab, because you can change water, you can change centrifugation conditions. So here we exploit a commercial device, and this comes from an interaction we have with a producer of membranes. I will come back here later. So these membranes in this cartridge is like a chromatography, let's say. So we can stack the graphene nanosheets on the surface of these fibers within this cartridge, because the cutoff of these membranes is much lower than the size of the sheets. And then we can wash the graphenes. Here we monitor the washing through the evolution of the fluorescence, because we bound for demonstration a dye on the PEI chains. And so then after having washed the graphene by the byproducts or unreacted species, you can recover the nanosheets by inversion of the flow through the cartridge, because we have 4 entry points in these cartridges. And so this allows us to avoid centrifugation and to get a standard method for purification, because these cartridges are commercially available and you can choose the size. We are also starting working with reduced graphene oxide. Reduced graphene oxide has less binding sites because it is reduced. So we have a PI system, much delocalized PI systems, but less defects, carbonylic and carboxylic defects to be modified. So here we exploited for modification a relation inspired by diazonium salts and diazonium reactions. But here we use these aryl azasulfones derivatives. The method was developed in collaboration with University of Bologna, Professor Bandini. So these compounds are thermally and thermally stable and photolabile in the visible range. So basically we activated this reaction by this homemade setup with blue lead, and we bound these aryl moieties that could be further modified by cross-coupling reactions to grow these brushes on precise sites of the reduced graphene oxide. But now, once we have this graphene and modified graphene oxide nanosheets, what we are trying to do is to use them in small amounts to integrate conventional treating systems. And I will mainly focus on these systems based on polysulfone and PVDF hollow fibers. These membranes are already used in filters that perform as ultra filter or micro filters for removing bacteria and viruses for water disinfection, but also for biomedical filtration, such as apheresis filtration, so plasma or blood filtration. So the idea is to exploit graphene to make, let's say, adsorptive membranes that are able to remove molecules, so emerging contaminants from water through absorption of graphene nanosheets, but also to preserve the filtration properties. And this is in mind the application of point-of-use devices or portable water purifiers, so small devices that require small-scale filters, and this is important because graphene is not ready to enter the market of municipal water filtration, but this could be a good entry point to take graphene to the market. So this is now a work in collaboration with membranes producers and also device producers in a framework of different projects, industrially focused projects. I'm deputy leader of or coordinator in the case of Flagera. So we have two ways to combine graphene and polysulfone or PVDF polymers. So basically coatings of these membranes or embedding of graphene in membranes. In this case, we patent, let's say, here I'm not showing the entire membranes. These are scraps of the production of the membranes, but the principle is the same. You can coat by the same method entire membranes or scraps of their production. And this idea comes from the interaction with these membranes producer. We discovered that they produce a lot of wastes, plastic wastes from the sizing of these membranes to fit the size of the commercial cartridges. So basically, we are talking about 10% of the total year production, ton scale per year of these wastes. So basically, we patent a method for grinding these scraps and to coat them with different nanomaterials to be used as sorbents, specific sorbents for several applications. This is now a project funded by the LIFE program just started. But the most industrially focused method at the moment for getting adsorptive membranes is based on phase inversion. Phase inversion is the standard method used by industries for making these membranes. Basically, you mix a polysulfone or the polymer with an organic solvent that's not so green. Let's say, I hope we will replace this solvent in the next years. You can mix graphene oxide in this mixture, and then when you put in contact this mixture with water, the polymer do precipitate and embed the graphene oxide nanosheets or the nanomaterials. So this method was used in our lab a few years ago, and this is the fabrication of a membrane in my lab in 2016. And today, this is the pilot line that the producer of these membranes working with us developed in the framework of the Graphene Flagship project. There's a dedicated spearhead, let's say, industrially focused project on this subject. And the pilot is implemented to commercialize these membranes and filters by the next year, so at the end of 2023. So you see here the hollow fiber spinning in the plant, and we are collecting the hollow fiber membranes with graphene. So here it's just to show you what does it mean an adsorptive membrane. This is a simple experiment. We mix a methylene blue dye with nanoparticles of PMMA of size higher than the cutoff of the membranes. And you see if you filter this solution through activated carbon, you remove the blue dye because the dye can be adsorbed by graphene by activated carbon. If you filter the solution through the polysulfone commercial fibers, you see that the solution is still blue because polysulfone can filter the nanoparticles but cannot absorb the dye. But if you filter this solution through polysulfone with graphene, you get both the absorption of methylene blue and the removal of the nanoparticles. So this is the working mechanism of the adsorptive membranes. And here we have only 3% of in weight of graphene oxide in this cartridge. So we are talking about few milligrams scale. We are aiding these companies to develop the best conditions to use graphene in this casting solution and in this industrial pilot. So basically here you see different cartridge with different amount of graphene oxide. The cartridge retain ultrafiltration properties. The cutoff is in the range of the virus size. And also absorb the molecules. This is again the standard antibiotic we use, and the absorption increases on increasing the amount of graphene, but we are stuck at a few percent, so maximum 5%. The safety of these filters is now under study in dedicated projects we are working with. We demonstrated the release below 0.1 ppb, which is the state-of-the-art limit of detection of graphene in water by a specific Raman spectroscopy, and a group of Professor Prato in Trieste is assessing the safety, the toxicity, ecotoxicity of these filters. And here you see a sketch of the working mechanisms. So basically through the porosity of the membranes, you stop the bacteria or viruses. It depends on the cutoff of the membranes, but graphene nanosheets are exposed to the surface of water passing through the membranes. And this is because during phase inversion, the nanosheets, graphene oxide is polar, is hydrophilic, so it migrates to the water phase. It is exposed to the surface, and so it can perform complexation or adsorption of emerging contaminants. And we are studying in more details the mechanisms and all the properties of these filters. And we are now validating them on a larger volume of water, tested through different pilots realized in collaboration with these companies. This is the suppliers of water in Emilia-Romagna region in Italy. We are also testing the filters in point-of-use devices. And also they are installed in a living lab in Sweden that is trying to reuse, purify and reuse gray water coming from the showers of students living in this lab. So here you will see our filters installed in this line together with the standard filters. So let me now jump to the conclusion. So this is an audience of chemists, and I have to say that we are so good in making chemicals, but we have to think on how to replace them with safer, in some cases, or with green molecules. And I'm talking in specifically of forever chemicals and disruptors such as PFOS, PFOA, or bisphenol A. That's a very bad story, an Italian story, but now it's a worldwide problem. When PFOS and PFOA, that are the most famous PFAS, were banned and they are now restricted, the company producing this compound that is very famous replaced them by two specific compounds and others that are more persistent and toxic than the previous one. And now in Italy, we have these compounds at the top in Veneto region and in Piemonte region. And also, I think that we have to put some efforts to develop these parallel systems for purification. And when I say parallel, I mean parallel to thermal separation processes that are now the standard method to separate chemicals. And this is an interesting Nature paper a few years ago showing us in the US, most of the energy consumption and carbon production comes from thermal separation, including most of them relating to chemical separation. So basically it suggests to develop specific membranes integrating nanomaterials on molecules that should promote specific separation processes. And I think this is a very point that we should take with a green mind, let's say. Now, let me thank the funding agency and projects that are funding our work. That's a very multidisciplinary activity. We are working in close contact with a lot of industries, producer of membranes or device for purification. So it's very important for us, their feedback and their suggestions, because sometimes we have solutions to problems that we don't know. And then I would like to thank my group, but only my co-workers in ISOF Institute and CNR in general, working at different level on synthesis, characterization, and water treatment. And of course, again, I would like to thank you, the division, and you all for your time. Thank you very much.
Thanks, Manuela, for your nice talk, for this very useful investigation for the solution of a very important problem. Now the discussion is open. We are late, so there is time for 1, 2 questions.
Can I ask a question?
Yes, sure, please.
How fast is the filtration with these filters?
In lab conditions, it's a few milliliters per minute, but we are targeting 3 bars. The top flow is about 3 to 5 liters per hours.
So there is a problem anyway for this.
Yeah, yes, we are now searching the, I mean, of course, the contact time is an issue, but we are now testing at these conditions the removal capability. Of course, it is lower because the contact time is quite lower than the time we use generally in for lab scale tests. But I know we have preliminary results. Of course, there is a drop of performance from, let's say, 80% to 20% removal. But the concentration of these contaminants in water, are trace contaminants, so few nanograms. We are also thinking to strategy to recirculate water, because you know the reverse osmosis as a tank you don't take water directly from the filter, but you recirculate and collect water in a tank, and then you have to drink this water within 24 hours. That could be the back strategy, let's say. So we are now working in parallel to be ready for failure in case the contact time it's not enough. But I'm aware of the problem. So.
Valeria, there's no further questions coming in from the participants that I can see anyhow.
There are other questions? No. So let me introduce the last speaker of this afternoon, Dr. Luca Dell'Amico, University of Padova. The title of his talk is Mechanistic Investigations in Light-Driven Synthetic Chemistry, Making Predictable the Unpredictable. Luca, the stage is yours.
Okay, thanks. Can you hear me well?
Yes.
Okay. So yeah, also I have to thank for the invitation. So I'm very honored to represent the Organic Chemistry Division of the Italian Chemical Society. And yeah, today I will tell you a story about what is going on in our labs in Padova. So first of all, let me say that in the group, we are working different topics. The first one deals with the design and synthesis and characterization of new photoorganocatalysts. Here are some recent reports from the group. So today I will not discuss about this type of chemistry, but I will focus more on the second research line that we are working with, so which is based on the development of new synthetic methods, so for the accessing of new potentially biological active substrate and the study of the mechanisms that are making these new materials. So I will mainly talk about unpublished results, but at first I will tell you how did we get there, so a bit of the story. So we see a lot of excited state carbonyl compounds, in particular aromatic ketones. So let me tell you very briefly that these are very special molecules because with respect to the aliphatic ones, they are much easier to use. First of all, we don't need a very strong UV light to promote their excitation, like in the case, for example, of acetone. And also the intersystem crossing to reach to the triple state is much more effective because the radical in the excited state, which lies in the pi star orbital, is in conjugation with the aryl groups. And this is not the case of aliphatic ketones. So we can work very easily with triple state of benzophenone and benzophenone derivatives. And if we think about a photochemical process involving carbonyl compounds, then we cannot forget about the Paterno-Bucchi reaction. So this is one of the more important, most famous reaction in photochemistry and in chemistry in general. And actually, the story started in 1908, when Emanuele Paterno discovered that shining light on ketones was possible to obtain more complex scaffolds, in particular oxetane. So this is a part of the original manuscript, so it is in Italian, as you can imagine. But I was also surprised how poetic was the writing of manuscript at that time. And let me also give you a brief overview of about what was the Italian Chemical Society at that time. This was the birthday of Stanislao Cannizzaro, was in Rome and was 1896. So we can recognize close to him Giacomo Ciamician, Paterno, also Biginelli was there. So it was really a golden age for the Italian chemistry society. Unfortunately, as you can see here, there's only one woman. So on this side, we have much improved. The second name in this reaction is the one of George Bucchi, which was a Swiss chemist and was working at the MIT. And in 1954, he first recognized what was the mechanism of this process, because there have been many discussions during the years. This chemistry was really fascinating for the scientists, and many different chemists proposed different reaction mechanisms, but was the first one that he realized that a biradical intermediate was involved in the mechanism of formation of the oxetane. So if we see the mechanism proposed by Bucchi, which is actually the one that is still accepted nowadays, so we can then shine light on the aromatic ketone. This will reach a triplet state, which has this biradical nature. And now we can trap this biradical with an olefin. So we can first generate the oxygen carbon bond. We will generate this stable biradical, which upon intersystem crossing and radical combination will lead to the final oxetane. So a very simple scaffold from very simple starting material. There are other mechanisms that have been discovered over the time, and I cannot avoid to recall the contribution of Turro, which envisioned an hexaplex between the triplet state, so an aggregation between the triplet state of the carbonyl compound and the olefin. And also Mattei realized that once the hexaplex is formed, we can have an electron transfer generating the radical anion of the benzophenone and the radical cation of the olefin. Quite recently, Greensbeck also was studying this intermediate, firstly reported by Bucchi, and he realized that this can rotate, giving rise to different stereoisomers. This, of course, can be a problem if you want to get the single stereoisomer, but it has an extreme potential if you think from a pharmaceutical perspective, because from the same reaction, we can get different type of products with different stereochemistry. So the synthetic potential of the Paterno-Bucchi reaction is impressive, because if you think how many times there are oxetane in biological active molecules, these are some examples, but there are really many other molecules which embody the oxetane scaffold. So, and now we have a very simple and cheap way to construct this complex system. Because if you think to construct an oxetane with moving 2 electrons, you may need 2 or 3 steps. But here you just mix light with a carbonyl compound and an olefin. So very cheap and very simple starting material to get these strained heterocycles. So during the years, the chemistry has been also explored with respect to peculiar double bonds. So we can use, for example, heteroaromatics. For example, we can dearomatize furans. So you can see here, we can generate the same biradical intermediates, and we can form these fused oxetane scaffold. And we also have the concomitant dearomatization of the furan. So in the group, what we were start to think about, it was, okay, so maybe we can take indole, which is a very important pharmacophoric course, which again is present in a lot of biological active molecules, to obtain its dearomatization with the formation of indolin, which is this blue scaffold that I highlighted in these molecules. Again, we are talking about important pharmacophoric course, and there were still at that time no report about dearomatization of oxindole using the Paterno-Bucchi process. So this was the idea. We take an indole, we take an aromatic ketone, and we shine light. So to start from a very simple planar scaffolds to get a complex 3D pharmacophoric course. Of course, there were many challenges to be addressed: the regioselectivity of the process, the stereoselectivity, the presence of side reaction, because, for example, it's known that this ketone can dimerize when you shine light over it, and also, of course, the indole reactivity, which was not reported before. So Pietro, it was PhD in the group, took care of the optimization of the process. So as you can imagine, the first trials met with failure, but then we understood that placing an electro-withdrawing group on the nitrogen and modulating the position 2 was the key. So we started to see the formation of the product 3. But one of the main problems was that we were generating a lot of this homodimer of the benzophenone 2. So this is coming from 2 excited state molecules of the benzophenone. So we start to reason how to reduce the formation of this side product. And we thought that the key was to reduce the concentration of the excited state of benzophenone, which is forming the product 4. And to do so, what we did, it was to move the emission wavelength of our lamp to 400 nanometers, which is at the border of absorption of the benzophenone. So we thought that using this wavelength, we were actually generating very few amount of the excited state, which will have encountered a larger exit of the indole. And indeed, this was the case. The reaction, of course, is lower, but for the first time in this entry, we observed the product for only in traces. Then we adjust the condition to 405 nanometers, and in this case, the productivity was the only product as a single stereoisomer. The reaction again was lower, but running the reaction in acetone, we managed to increase the reaction time. We were very surprised that we were able to use visible light for the excitation of benzophenone. So this was the first report after the study of this reaction. And so we first, together with Francesco, first overimposed the emission of our LED with the absorption of the benzophenone. So you can see that there is a region in this where these two spectra are overimposed. And if we run a 3D matrix experiment, here we have the excitation wavelength, here we have the emission of the triplet state. So we can see that actually also at 405 nanometer, we are generating enough excited state to result in this emission here. And if we move to 365 nanometer, then we roughly generate the double amount of excited state. And this is why we observe the homodimer at these more energetic wavelengths. Then with Javier and Alberto, we performed the scope of the reaction. I don't want to be annoying, so the reaction is very general. So you can vary position 2 and position 3, and also the indole scaffold. You can place different protecting group. One of the only limitation is that you need an electro-withdrawing group on the nitrogen, as I was mentioning before. And also the reaction is general with respect to the carbonyl compounds. You can use different benzophenone. You can use prochiral benzyl derivatives. In this case, we generate 3 contiguous stereocenter with full diastereo control using visible light. So we next wanted to see if it was possible to use this process for the construction of the complex pharmacophoric cores I showed you before. And indeed, this was possible, and we accessed these complex tetracyclic scaffolds in quite good yield and very high diastereo control. Also, we were able to make reaction with melatonin, tryptophan, and tryptamine. Also, these substrates were used under our process. We overcome the limitation of the electro-withdrawing group on the nitrogen with a post-functionalization. We can, of course, reduce the bulk to the methyl group by using lithium aluminum hydride. And next, we also implement this process starting from oxindoles. In this case, the key was to in situ generate the corresponding indole. So with the use of a base and a different protecting group on the nitrogen, we were generating in situ the indole, which was trapped through a Paterno-Bucchi reaction by the excited state benzophenone with the formation of the oxetane system. This reaction was performing in flow, and the reaction was much faster, and this also allowed us to access gram scale of product. So I don't have to explain you more about flow chemistry because Renzo already showed you different setup. So also in this case, the reaction was very general, but as you can see from the scope, I'm not showing you prochiral carbonyl compounds, and I will go back to this in a moment. So we designed this cover together with Eduardo was a master in the group. He painted on his own hand this picture inspired by this creation of the birds, which was a painting by Remedio Varos, a Mexican-Spanish painter. So we can see here the microfluidic conduits together with the aid of light are making real this scaffold by this birdman, which is an alchemist. So you can see here the actual X-ray of the molecule, which is extremely strange. So you can see how hindered is this scaffold. Here is the oxetane, and about 90 degrees, we see the plane of the indole. Okay, then at that time, we wanted to try also the reaction with this electron-rich indole with the prochiral derivatives, and we tried to use benzyl in this type of chemistry. So Ana Javier and Philip tried this reaction at 465 nanometer, but they got moderate yield and poor DR. So we were discussing about this, and we thought, okay, let's try to increase, to use a more powerful wavelength. Let's go to 370 nanometer. even if we knew it was possible to observe the homodimer of 2A, but we thought maybe we will increase the yield anyway. And this is what happened. But together with this, we observed that the DR was changing. Actually, the reaction was more endoselective. So we start to think about what is the effect of the light on this process. And on the other hand, we also set up the reaction with the alpha ketoester. And also in this case, when we start with the methyl group, we got a low yield and also quite low DR. And then what you usually suggest to students, like, okay, take something bulkier, then the DR will improve. But what happened here was exactly the opposite. So we switched the DR. Now the major compound was the exo product when we moved to the isopropyl. So again, we have another question to answer. What is doing sterics in this condition, in this reaction? So we decided, we are very curious, and we decided to go deeper and study the mechanism of this process. And together with Mirko Natali from Ferrara and Javier, we started to study the process with simpler scaffolds. So we went back to benzophenone and this 2-methylindole. So, and we performed transit absorption spectroscopy to observe the formation of the radical intermediates. So this is a classical Paterno-Bucchi process, the mechanism that actually was discovered by Bucchi. So we generate the triplet state, and now we trap the triplet state with this double bond of the indole. We first generate the oxygen-carbon bond here. We do see by TAS the 1,4 biradical, which correspond to this absorption. So while the triplet state is going down, the triplet biradical is formed, and then this is generating the final product. Then when we move to more electrophilic benzophenone, the chemistry is changing significantly. So we have two alternative pathways. If we are under diluted condition, we are generating an exciplex like the one that I showed you before. We was studied by Turo and Matai. So here we have the triplet state, which is aggregating with the ground state of the indole, and this is evolving to a radical ion pair upon electron transfer. So the indole is giving one electron to the excited state, generating the radical anion of the benzophenone, the radical cation of the indole. So now we do not observe the formation of the 1,4 biradical here, but we do observe upon decreasing of the triplet state, the generation of the biradical here. When we are under more concentrated condition, we do observe the aggregation at the ground state of the two reagents. So in this case, we have what is called an electron donor acceptor complex, an EDA complex. So we have an aggregation between the electron poor ketone and the electron rich indole. So we can trace this by titration UV titration experiment. We do observe the formation of a charge transfer band. If we irradiate this charge transfer band, this complex is evolving directly to the charge transfer state. Now we do observe very few amount of the triplet state together with a large amount of the charge transfer state of the biradical system. Both these reaction pathway are leading to the same product 3B, but in a different manner. So with Paolo Costa, we decided to gain insights into the arrangement of these two different manifold, because this can control the geometry of the final oxetane. So in the exciplex, these are minimized structures, so we can see that we have this interaction between the triplet state and the indole. These are the DFT calculation. We run spin density map. This tells where is the radical. And you can see that this radical here is delocalized over the whole aromatic system of the benzophenone, sorry. And the other radical is actually lying on the aromatic core of the indole. So we have a system in which the radical is all around the molecule. And if we relax this system now, what we observe, here we see the charge distribution. So basically what happened is that the indole has given one electron to the triplet excited state. Basically we have the radical cation and the radical anion of the benzophenone. On the other end, by the EDA complex, as you can expect, the LUMO is localized on the electron poor benzophenone, while the HOMO is localized on the electron rich indole. The system is almost neutral. We have very few dipole moment, but again, upon excitation, we can see now the charge distribution is changing very much. So basically the indole has given one electron to the ground state benzophenone, generating its radical anion. So we start thinking, so maybe we can develop a light diastereodifferentiation process, because if we shine light on the single ketone, so if we excite with UV light the ketone, we are promoting the formation of the triplet state. We generate an exciplex, which has this different geometry, which is evolving to a charge transfer state, keeping this geometry here. On the other hand, if we have an EDA complex, we have the aggregation at the ground state, and this is leading to a different charge transfer state. So we may be able, by switching the light, to switch the stereochemistry of the product. But in this case, of course, we need a prochiral carbonyl compounds. So, and that's why we use benzyl. So in this case, we can envision two different transition state, two different reaction manifolds, one which is based on the exciplex, which leads to the endo product, and one which is based on an EDA complex, which leads to the exo product. And we first proved the formation of an EDA between the two reagents. So you can see at the titration here, we generate a new charge transfer band, which indicates that we are forming an EDA complex under concentrated condition. Then we start our experiment under diluted condition. The reaction is fully endoselective. But when we move to the visible light, so we start to see the formation of the exo product. This is because, of course, we are not irradiating anymore the single benzyl, but we are exciting this EDA complex. And if we run the reaction under more concentrated condition, the exo product is more is observed in higher concentration, because we are favoring the formation of the EDA complex. And finally, running the reaction at lower temperature, we stabilize both pathways. And here you can see that we can obtain the full endo control when we use 370 nanometer, and we get about 1-to-1 DR when we move to the visible light. So by switching the light, we can control the stereochemistry of the product. And we wanted to prove that also using, to confirm this hypothesis, an aromatic solvent such as benzene or toluene, you destroy this interaction in the AD complex. And indeed, when we use benzene, we did observe a reduced exoselectivity. We then move to prochiral alpha ketoester. We have the two same possible reaction manifold, but here the situation is a bit more complex. So we first study the electronics. So we can place electron donating groups and electron withdrawing groups. So you can see that the DR basically is not changing. We also have roughly about 70-30 DR in favor of the endo, but the only one that show a different behavior is this BCF3 ketone here. And indeed, if you plot the DR versus the reduction potential, we do observe a variation in this graph. And this is because this is the only ketone which is able to generate an EDA complex with the ground state indole. And that's why we observe this different behavior. The reaction is much more endoselective. So we next screen the steric factors playing with this R group. So we can see that the reaction is very endoselective for both when we have the phenyl and when we have the BCF3. But when we have the BCF3, this the astero switch that we observe when moving to bigger groups here is higher. The sensitivity is higher because, of course, in this case, we have an EDA complex. So what is happening here? That we are increasing the size of this R group, and then we are disfavoring this type of aggregation. And if the R group is bulkier and bulkier, then the system goes through this exciplex transition state where the S group is pointing outwards. And then we start to observe the exo. Actually, here passing from methyl to isopropyl, we already observe a switch in the diastereo control. And then what we did, it was to play a bit with the temperature again. So when we place the methyl group, we are favoring the EDA complex. We get about 90-10 DR. But when we move simply from methyl to isopropyl, we switch to DR, we get 85% of the exo product. So just changing this group on the oxygen. So this is the summary, and I think now it's easier to understand something that at the beginning, at least for us, was really unpredictable, because now we can play with different indoles here. So we can place an electro-donating group or an electro-withdrawing group, but you can see the behavior is very general. So we can move from the endoselectivity when we use UV lights to the exoselectivity when we pass to the visible light, and we can completely reverse the selectivity playing with the sterics. When we have an isopropyl, the reaction is exoselective. When we have a smaller group, we stabilize the EDA complex transition state, and then the reaction is fully endoselective. Okay, this is my last scientific slide. So many of you know where is Padova, so I will not comment too much, but it's a beautiful Renaissance city. So if you didn't come, you are welcome to come here, the city is very close to Venice. It's actually connected by canals to Venice. And let me say that this year we celebrate 800 years of our university, which is one of the oldest universities that we have in Europe and in the world. So this is the most important slide. So all the people that did the work, all my collaborators over the time, the foundings. And of course, let me thank all of you for your attention.
Thanks, Luca. Very nice talk. There is time for one question. It's very late, so please ask. The discussion is open.
So Luca, that's an excellent presentation. I really enjoyed it, and it shows you have a lot of light in Italy and you're using it very well in your chemistry.
Thanks.
Very high levels of endo versus stereoselectivity, but will you ever solve the problem of enantioselectivity?
Yeah, this is very challenging. We have tried something also before we understood deeply the mechanism, and we got very few results. So we didn't manage to have high enantio control. But I think now that we have the full mechanistic picture, we have more chances to control, and this is what we are working on. So thanks.
It's funny, I just looked at the questions and answer after I asked that question, because Valentina Giraldi has just said, hi, Prof. Dell'Amico, have you ever tried to do an enantioselective Paterno-Bucchi reaction? So great minds think alike. There's another question from Bartosz Bieszczak, who's a former PhD student actually here from Dublin. Very nice chemistry. Can I ask how technically did you carry out photochemical reactions at -78 degrees Celsius?
Yeah, I mean, we prepare a bath and we basically place the irradiation source on the top of the bath, and this was enough to drive the chemistry. So nothing special. Also, the reaction was quite fast. So in 2 hours, the reaction is finished. So we don't need to maintain the bath for a long time. Otherwise, you need a cryostat, of course, to maintain the temperature down.
Okay, I don't see any further questions.
No other questions.
Okay, if not, perhaps if I could maybe wrap up. What a fantastic afternoon of chemistry. I have to say, I really enjoyed it. It was really superb, and very many thanks to the President Valeria Dauria for her support of this initiative, and of course to my colleague on the Division of Organic Chemistry, Gianluca, for his support of this as well, and for suggesting that maybe Italy could go first. So that was great, and hopefully other colleagues in the division will see how successful a day it is for their country, and maybe they will likewise take up the baton, so to speak. And just for your information, the 10th of June will be highlighting organic chemistry in Ireland, and with a bit of luck, the third initiative will be in with Finland, with Ari Kostinen. So we'll see how that all works. Valeria, many thanks for that superb introduction about the importance of chemistry and the role that chemistry plays in Italy. I mean, one of the things and why I came up with this initiative is that we know vaguely some things that are happening in other countries, but we don't know the detail. And I think it's really nice to find out about the range of courses that are available in chemistry in Italy, the vast range of universities, the numbers of PhDs, the sort of research focus that there is within Italy, but also the importance of the chemical industry to the country. You know, I mean, I learned about the 10th most important country for production. And it'll be interesting as we go around the various countries and societies in Europe as to how many other people say they're the top of this or top of that or wherever they are. So we'll get a flavor about the importance of organic chemistry in each of the countries across Europe as we do that tour. 1 every 3 months, that's the plan. So, you know, it's a nice way of doing it. However, the main success of the afternoon really has been the standard of the 7 talks. They were really superb. So, you know, many thanks on behalf of EuChemS for giving up your time. There were really superb presentations across a breadth of range of things. You know, we had Maurizio Prato talking to us about nanodots, and I did notice that your microscale to nanoscale ended up at a tennis ball. Maybe that's because Matteo Berrettini and Yannick Sinner are such good tennis players at the moment. But I did notice it didn't show the size of a soccer ball. Now, that might be the slide might have changed between yesterday and today, because of course, there was a very shock result last night in injury time where North Macedonia unfortunately beat Italy. That was a shock, I think, for the soccer world. What lengths you would go to not go to Qatar. Ireland, by the way, we just played badly in our group, but we didn't go anywhere either. So we're not going to be in the World Cup for that reason either. So really, there was superb talks all the way. I enjoyed it. Many thanks to Flaviana. Again, we use LPS in a lot of our into anti-inflammatory type work, and I've never really thought about why we add in LPS and what exactly it does. So yours is a really interesting talk. I know Walter so well in terms of his palladium chemistry, and I thought there was a very insightful talk in terms of the importance of green chemistry and why we should think about solvents and byproducts a little bit more carefully than perhaps we do in the first place. And then just before a very short coffee break, we had Anna who talked about really the importance of glycomimetics and charged species and all of that. So it was really superb. Then we had some flow chemistry from Renzo, again, really nice and making small rings, big rings and adding them on. Manuela, then, as we all know, it's important to have clean water, but we don't often think about exactly how we might get about getting clean water. And that whole green sustainable goals was really superb presentation. Again, I would say of not just modern, but future chemistry and what we should be looking at. And then we finished up with a superb talk again on photocatalysis. And of course, Italy has been one of the leading countries for many years because it has such a great amount of sun in photochemistry. So really, it was a superb afternoon. I thank you all, and I thank all of the attendees. And just to give you some numbers, we had about 440 people registered. At our optimal, we had about 320 people listening in this afternoon. And unsurprisingly, on a Friday afternoon, it attenuated somewhat, and we're at about 85 now. But look, that's fine. That just shows that there is an appetite for afternoons of chemistry like this. You'll have noticed if you look through the attendees that unsurprisingly, there was a lot of Italian attendees. There were a lot of Irish attending as well, actually. So I think as we go around the various countries, hopefully we'll get an equal distribution. Obviously, there'll be a huge number from the country involved because they're the country being highlighted. So again, a superb presentation from all of you. I really enjoyed it. This has been a great start. I hope you agree, Gianluca and Valeria, for the initiative. And as we work our way across Europe, up and down, north and south, east and west, hopefully we'll all learn a lot more about each other, because that's really what this whole thing is about. Because, the Division of Organic Chemistry and EuChemS has good oversight of what some people do, but we didn't know the details. So hopefully with more of these days, we will know more about what's going on. So again, from Dublin on the very west side of Europe, I thank you for giving your time and efforts to this. Again, superb organization, Valeria and Gianluca, thank you very much. And as Europeans, and I noticed some Ukraine flags in some of the talks, our thoughts go out to our colleagues and chemists and researchers and the people of Ukraine during these troubled times. So thank you for your time, and I wish you all a very lovely weekend. So grazie mille, grazie, I should say. Gaurav Mila Mahagut in Irish. Thank you very much.
Thanks.
Thank you very much.
Thank you very much. Bye-bye.
Bye. Thank you.
Bye.
Okay, bye, folks. Now.
Grazie Valeria.
Arrivederci.
Grazie, arrivederci.
Grazie, arrivederci.